For those of you who are typing in the chat box, not audible, we are just waiting for Dr. Shirsagar and Dr. Jayaprakash to join. Please hold on. We are running a few minutes late, but we hope to be able to start soon. Thank you, everybody, for your patience. Good afternoon, everybody. I'm trying to reach Dr. Shirsagar as well as the first speaker, Dr. Davis. Uh, I'm not able to connect with them. I think we'll give it another 5 minutes and get started by 1:45. My sincere apologies for the delay.
On behalf of the organizing committee, I, Dr. Pallavi, Assistant Professor from Department of Clinical Pharmacology, wish you all a very good afternoon and extend a warm welcome to the advanced level training workshop in pharmacovigilance under the aegis of PVPI. At the outset, we would like to thank our Dean, Dr. Sangeeta Rawat ma'am, for giving us the opportunity and providing us the support to conduct this workshop. We also want to extend our gratitude to the heads of departments, Dr. Nadkar, HOD Department of Medicine, Dr. Karande, HOD Department of Pediatrics, Dr. Chandrakala, HOD Department of Hematology, for their continuing help and guidance towards the pharmacovigilance program at our center. We believe that the upcoming sessions will take you through some domains in pharmacovigilance and enhance your perspective on them. Before we begin, there are certain housekeeping announcements. Please keep yourselves mute at all times during the workshop. All questions from the participants should only be typed in the chat box. And thirdly, the questions will be moderated by the respective chairperson at the end of each session. Now, I request Dr. Mahesh Belekar, Associate Professor, Department of Clinical Pharmacology, KEM, to kindly give a brief introduction to the workshop and formally welcome everyone.
Good afternoon. Thank you, Dr. Pallavi. Good afternoon, all of you. I'm Dr. Mahesh Belekar. On behalf of our Department of Clinical Pharmacy at GS Medical College and KM Hospital, Mumbai, and Pharmacovigilance Program of India, I welcome all the chairpersons, speakers, and delegates for today's 3-hour duration advanced level training workshop entitled as Emerging Themes in Pharmacovigilance. I would like to mention here that our department is an ADR monitoring center, that is AMC, as well as a regional training center, that is a RTC, under the Pharmacovigilance Program of India. And as per the mandate of Pharmacovigilance Program of India, we have conducted a similar type of advanced level training workshop in the month of February 2022. And today's workshop is our second workshop of this year. In today's workshop, we have well-known speakers, and the agenda for today's workshop is displayed on this slide. First, we have talks by today. We have first talks by introduction by Mr. Dr. Denis Xavier, and followed by talks by Dr. Nitya Madam, and followed by on artificial intelligence. So, with this introductory remark, I request Dr. Pallavi, our assistant professor, to proceed with the comparing. Thank you.
Thank you, sir. It's indeed a privilege for me to compare the workshop. So we first start with the session on artificial intelligence. As we move ahead to it, I invite Dr.
Pallavi.
Dennis.
Pallavi, we
Yes,
have
ma'am.
Dr. Jayaprakash from IPC.
Yes, ma'am. I invite Dr. Jayaprakash, sir, Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India, Ghaziabad. Sir has completed his doctorate from Department of Pharmacology, All India Institute of Medical Sciences, New Delhi, India. Sir, in his illustrious career, has done commendable work for more than 20 years in the area of teaching, research, pharmacovigilance, pharmacopoeia, and formulary sciences. He is the member of National Expert Committee for Allergens, Institutional Health Ethics Committee, and National Institute of Biologics, Noida. Sir has been closely associated with our department for many years. It is pertinent to mention here that the pharmacovigilance program in India has flourished in abundance under his able guidance. I request you, sir, to give us the IPC perspective through your presentation.
Thank you, Dr. Pallavi. All the respected participants, especially Dr. Nitya Gogte, the coordinator of this AMC, who has been shouldering the responsibility for efficiently running the AMC as well as the regional training center of the PVPI. So at the outset, I congratulate Dr. Nitya Gupte and her team for organizing this important advanced level training program on pharmacovigilance. The KEM hospital at Mumbai has been the AMC of under PVPI since 2011 to best of my knowledge, and since that time it is also designated as the regional resource and training center of the PVPI. This center has done very good work in submission of individual case safety reports from the western region to the PVPI, and we have found out the data from the PVPI database that they have submitted around 2,500 ICSRs to Pharmacovigilance Program of India since their AMC was established, and just in the year 2022, more than 1,100 ICSRs have been submitted by them, and they have taken active interest in organizing the training programs. 22 training programs they have organized, and 6 trainings they have conducted during the National Pharmacovigilance Week. There is a lot of responsibilities on the institute as well as on the shoulders of the coordinators, deputy coordinators of the regional resource training center, and I am confident that they will continue to support for the national cause of submission of the ICSRs to the PVPI, and will keep the image of the Pharmacovigilance Program of India at the highest level. And in the recently during the core training panel meeting of the PVPI, it was also decided that each of the regional resource training centers has also to coordinate with the public health programs in their region, as well as with the Indian Medical Association branches also in their states. And I request Madam to kindly take note of these things also, that coordination with the national health programs as well as IMA units will also boost the public health programs as well as the Pharmacovigilance Program of India. And I have seen the topics which have been selected by Dr. Nitya Bhogte. They are the contemporary as well as the advanced level topics in the area of pharmacovigilance. So with these words, I extend all my best wishes for the success of this advanced level training program. Thank you, everyone. I think everyone will enjoy with the interactive question answer session also once each topic is completed, and they are also free to ask the questions either through email or by other communications also as convenient to the coordinator, and they will be very happy to reply to the questions of the audience. Thank you, everyone. And I also look forward to listen to the presentation
Nitya,
of
good morning,
the event speakers who are here.
Can you hear me?
Madam has also joined, so we welcome.
Am I audible?
Yes, madam. Yes, madam. Yes, madam. You're audible.
I can't
We
hear
also
you. Just a
we
minute.
welcome our chairperson of the core training panel, Dr. Neelma Chirsagar. Madam has been supporting a lot as and when her help is required. We feel free to contact her anytime, any moment, in the interest of patient safety and pharmacovigilance program of India. Madam, welcome to you. And I again extend my best wishes to the success of this advanced level training program. Thank you, everyone.
Dr. Jayaprakash, thank you very much. Madam, thank you for joining. Is it clear, audible? Are you able to hear us?
Yes, I can hear you. Can you hear me?
Yeah, madam, very well. I'll just ask Palvi to introduce you. We missed you earlier. We started at 1:30, madam, India time. So Palvi is here. So we had Dr. Jayaprakash's talk ahead of yours. Yeah.
Yeah, sorry, I got the
No, no.
timing a bit
No problem.
mixed up.
Yeah, Palvi, over to you. Yeah.
Yes, ma'am. I welcome Dr. Nilima Sheesagar, ma'am. Ma'am currently holds the position of Chairperson, Core Training Panel, PVPI. While everyone knows about ma'am, I would like to speak briefly about her credentials. The Department of Clinical Pharmacology at KEM was started by her in the year 1995, formerly holding the post of the head of the department and also serving as Dean KEM Hospital and Nair Hospital. Currently, ma'am is Emeritus Scientist, Chairperson, SAG BMS, member Scientific Advisory Board ICMR, and also member DTAB, Chairperson FDC Subcommittee, Government of India. Ma'am has also held the position of National Chair, Clinical Pharmacology ICMR in the past. With her strong presence in pharmacovigilance, she was instrumental in originally starting pharmacovigilance in our institute and our department as well. I request ma'am to kindly give a talk of an introduction to the core training panel.
Thank you very much. Thank you, Pallavi, for that introduction. Some of these positions are now former positions, though. And thank you, Ditya, for inviting me for this very important session that you've organized, advanced training program. And thank you, Jayaprakash, for all the support on the training panel that you provide. I can see that there are a number of participants. The core training panel has multiple components of it, and which I'm sure Jayaprakash would have already elaborated on. Apologies for joining a bit late. I'm in UK, and I think I miscalculated the time gap. But I would just like to mention here that we have a very large number of advanced, we have very large number of ADR monitoring centers. And then we have regional centers, such as the one with Dr. Nityan at KEM Hospital. And today, while we are having the program, which is advanced level program, I would just like to mention a few points here, particularly in light of the issues that are happening around. Some of these are very well known and very common, but nevertheless, you will see how important it is that the pharmacovigilance, all those who are working in pharmacovigilance understand the importance of pharmacovigilance and the importance of, I would use the word case narrative, and also the importance of being able to exactly state what are the possible or potential likely drugs or medicines which are likely to cause this particular area. So recently you might have read in the newspapers also about the uh, possible cough syrup causing deaths in Ghana. And I was looking at some of the WhatsApp messages recently, and there was another cough syrup which was being considered as a possibility of when a child got nearly unconscious with the cough syrup. Now, here I would just like to reiterate we are going to have a extremely interesting and important sessions in the coming few minutes and hour. But in Ghana, as we can see from the newspaper messages that come, the case narrative and therefore the causality assessment could not be done very appropriately, and also which drugs were given. So I like to mention this to everybody that whenever you are involved in any case report, this is something which is crucially important. Recently, we were looking at public health related uh, use of a particular medicine which was causing probably adverse reaction which was being evaluated. And what we noted was that the information was really incomplete. So I would urge everybody that when you get any report of any ADR, you must personally look into, not depend only on what the prescriber or the reporter has provided with the information. You need to ensure that you get into all the details of the adverse reaction. The causality assessment, which is we all follow WHO causality assessment of certain probable, possible, and then unclassifiable or unclassified and unlikely. So here, one of the important things is to note whether the disease itself or any other factor could have caused this particular adverse reaction. Now, for that to understand very well, you need to collaborate or have the confidence of the clinician who are treating this patient, need to know a bit more about the disease process itself, and then be able to assess the causality appropriately. So we are having later on artificial intelligence as also being a talk which Dr. Nitya is giving. And I'm sure that is something which is a very important aspect of pharmacovigilance now, and that you're all getting involved into this particular area as well. So that is something which will come along in some time in future. And we also have some very interesting other aspects from Sanish and others. The last point, therefore, I would like to mention here is that whether we have artificial intelligence or howsoever well the pharmacovigilance program itself collects all the information, ultimately it is the communication to the prescriber, the patient, and general public that's very important. And I'm sure that is another area which needs a lot of emphasis. So the pharmacovigilance program of India has in the past couple of years has taken a lot of initiative, and as Jayaprakash might have mentioned, this is also the WHO collaborating center. So I think all those who are present here should make full use of all this in enhancing their own knowledge and ultimately providing to the safe use of medicines. Important in pharmacovigilance is there to be able to do the risk management and be able to prevent. So to be able to again assess the risks which need to, so that the prescription is such that you reduce the risk to a particular medicine. This also can be assessed only if the case narrative and the case description is appropriately given. ICMR has recently now, we had a rational use of medicine centers, and now the next 3 years going to be safe and rational use of medicines. So I do hope that in next 3 years that this particular activity of ICMR gets also well connected with the pharmacovigilance program, so that we are able to collectively as a scientific community provide the best to the patients, so that we are able to really prevent ADRs which are known and able to detect early the ones which are unknown sort of to us till now. So with these few words, I once again thank Nitya and good best wishes for a very successful this workshop. Thank you, Nitya. Thank you very much.
Thank you, ma'am. Now we move towards the first session for the day. I invite Dr. Sheesagar ma'am as the chair of the session. I request you, ma'am, to kindly introduce the speakers and take the proceedings forward.
Ma'am, you're muted.
Are you going to put this up on the screen, or have I
No,
to
madam.
look up? No, you're not going to. Okay, then give me a minute. I'll just look up onto the message that you've sent. Sorry.
Yeah. First speaker is our own Sanish, madam.
Yeah. So first is I'm looking on my mobile. Not sent me on the mobile. Okay, if it is Sanish Davis, then I don't really need to look at any CV of his. Is one of the most brilliant, outstanding clinical pharmacologists. He has done his training here in KEM hospital, and we are all very proud of all his achievement. He's also the president just now of the ICSR. And through that, he has conducted some very effective and important training programs and workshops, which are critically important, such as of multi-regional clinical trials and so on. And he has held many important positions in industry, and right now also he heads a very important medical director's position. So Sanish, I'm sorry, that's not a full introduction or CV of yours, but all of you know him. Thank you, Sanish, and request you to go ahead with your presentation, please.
Thank you so much, Shivsagar ma'am, for your very eloquent introduction. Really appreciate that. And thank you, Dr. Nitya and other members of the organizing committee for the opportunity to present on something which is a little bit different from the standard pharmacovigilance sessions, which maybe people who have joined this program might expect. But I'll try to see how to link this all back in. But maybe before I actually start on my talk today, I want to again reiterate what Dr. Shivsagar said. Sometimes the start of, you could say, a very big important safety issue would have been with, you could say, the simple reporting of that in the most appropriate way, right? We have all been trained to actually report an adverse event in the proper way. Oftentimes, the improper reporting of this actually makes issues much bigger and much larger than how it is perceived, you could say, at the ground level. So thank you, ma'am, for always bringing us back to that most important, you could say, lesson of how to report an adverse event appropriately. So with that, my customary disclaimers that this is a presentation which is done in my personal capacity, and there are several excellent manuscripts or articles on immuno-oncology, immunotherapy, etc., and they can be referred for further information. I would also want to actually provide a brief overview of what I'm going to cover today. A preliminary account of the immunotherapy agents. I also know that the next speaker is going to talk more about CAR-T. So this session will certainly segue well into that particular section on CAR-T. We'll talk about some preliminary considerations of toxicity and by extension of toxicity, I would also mean the safety parameters that needs to be looked at for immunotherapy agents. And you could say the safety of a compound actually starts all the way from its development into when it is marketed, right? So I will talk a little bit about the early clinical development of immunotherapy agents and how it is a bit different from a safety and a toxicity profile in the early stage development. And also will, because this is done in clinical pharmacology department, I'm sure the audience would also be interested to look at what are some of the different types of considerations for early clinical development, especially in phase 1, that some of these agents have to actually look at. And then last, I'm going to link it up with the operational part of how safety is reported in clinical trials with these particular agents. And it's very interesting that while KEM is a reporting center, we would also need to actually have an eye on social media and other places, because sometimes clinicians and investigators sometimes first reach out to social media to actually report an adverse event or a serious adverse event. So some of those interesting parameters and perspectives will also be brought in. So with that, I think immunotherapy, as many of you would know, has literally changed the way in which cancers are now being treated. These are extremely costly drugs. That's something which I would like to state upfront. But many of these are like the logical progression of right from immune checkpoint inhibitors, monoclonal antibodies, cancer vaccine, and now, of course, the latest addition to that clinical artillery, which is in the form of the chimeric antigen receptor or the CAR T cell therapy, which the next speaker will talk about extensively, and the Indian experience, especially on the safety side. When we were actually looking at whether India can do some of these studies, there were a lot of questions that were asked by global companies or people, clinical development folks in global organizations, that India does not have too much of experience with many of these situations, like, for example, cytokine release syndrome, right? Many of us would now know post-COVID that cytokine release syndrome is something which is a common occurrence which happen in COVID as well as with treatment with many other situations. But several years back, before COVID, CRS was not a term which was commonly heard in the clinical practice, as well as in ICUs, as well as in critical care specialists, but is a very common adverse effect that is seen for several monoclonal antibodies which treat a plethora of targets. And sometimes it's also associated with neurotoxicity, which kind of like complicates the whole situation. And of course, this is also much more common and more severe with CAR T therapy. And I would be interested to hear the Indian experience from the next speaker on this too. Both of them require prompt recognition and treatment. And of course, in India, during the COVID period, especially patients were having a CRS episode, the availability of tocilizumab, both the innovator compound as well as several generic versions of the compound, was really a boon for many patients who actually had a cytokine release syndrome or who could be treated for these kind of situations. While CRS and neurotoxicity are very specific toxicity that is associated with immunotherapy, there are very niche adverse events also seen, or toxicities that are also seen with these particular agents. And I would like to just call out the sinusoidal obstruction syndrome, which sometimes is very fatal, that even 80% of patients who are treated with this particular antibody drug conjugate can actually have even multi-organ failure, etc. Now, shifting our focus to, you could say, the early clinical development side of things. And we actually talk about a regulation which the US FDA brought out, which is actually leading drug development in oncology at a very fast pace. I mean, that's a pathway that every company is now moving their drugs into. So this particular regulation or this particular pathway is called as the breakthrough designation pathway. And this just means that if you have a drug or a compound which shows initial clinical evidence on a clinically important or a significant endpoint, and that shows that there is substantial improvement over other existing therapies, then you have a potential for a drug to be designated as a PTD. And such definition or such designation by the US FDA overall accelerates your pace of your new drug development, or you could say it kind of like puts you in a pathway which is like you're suddenly you're upgraded from 100, you could say, from a marathon into a 100-meter race. I mean, that's how fast the pace of development then happens. The first drug that got through this particular designation pathway was pembrolizumab. The compound name is well known to many people. It can treat a plethora of cancers, but the first approval came through for melanoma in December of 2014, and that was based on a phase 1 study, right? And it was a dose comparing activity estimating cohort within a open label phase 1b study. I mean, that's the study that was there. This also used an objective response rate as one of the key endpoints that were there. While the approval was given, of course, the US FDA also committed the company to actually conduct then a phase 3 superiority design versus the SOC. But just imagine, clinical trial, that to a phase 1 study is the one which led to the drug's approval for marketing. And now we know post 2014, it's now close to 8 years, and pembrolizumab is a drug which is used for treating a plethora of cancers, not just for melanoma. Now, from there, if you look at the current way in which the scientific thought process is going for oncology drug development, the thought process has always been that the more toxic a treatment is, the potential for it to be more efficacious or more, you could say, useful was there. So every oncologist would actually, who was into drug development, would say, the most important goals was to define the maximum tolerable dose, or the MTD, and also to limit the number of dose limiting toxicities for your compound. If you're able to strike a fine balance with the MTD and the DLTs, then you have a potential drug which can move into drug development and ultimately get into an approved drug. But increasingly, with the monoclonal antibodies coming into being, there is this third goal which also has started to gain a lot of traction right from 2010, etc., which is to find the optimal biologically active dose level and to gain experience of both efficacy and safety at that particular recommended dose level. So that's how you could say the third goal would also be something that would be used. Traditional oncology phase 1 studies, and these are mainly for chemotherapy agents, and now increasingly there is a change from the traditional 3+3 design, was to find out the MTD and the DLTs at the same time. And many of you who are part of clinical pharmacology, etc., would know this algorithm as to how this is done. But this traditional approach to finding the dose limiting toxicities and the maximum tolerable dose was initially propounded mainly for the cytotoxic chemotherapy agents. But with the advent of newer agents like immunotherapy agents, there are a lot of challenges or a lot of issues with regards to using or following this pathway. And now there is a talk about the need for amending these particular, you could say, development pathways or this traditional approach that is there, and which also puts a lot of emphasis on how statistical considerations are going to be used for these particular agents. Now, what is different for immunotherapy can be put in these 4 different buckets. The first one is the appearance of late-stage toxicity with these agents. Now, traditionally in phase 1 clinical trials with chemotherapy agents or with existing drugs, the dose-limiting toxicities are usually looked at in the initial cycle when one of the treatment, and which is typically around 28 days long. And the MTD is defined as the maximum tolerated or the highest tolerated dose from this cycle, even though we know that oncology is not just about one cycle, but there are multiple cycles that the patient has to go through depending upon which therapeutic area or which indication it is. So it was appropriate for cytotoxic drugs because you could see the DLT and then find the MTD, etc. very early. But this does not, you could say, lend very well for immuno-oncology agents because these agents have toxicities which come up after an extended period of time. And these are called as the immune-related adverse events or IRAEs. And it is very difficult to actually see that within the first cycle, you'll be able to find out what the, you could say, the DLTs and others are. So there have been several proposals as to one, increase the DLT timeline window, that instead of just looking at it in cycle 1, maybe look at maybe a couple of cycles. There have also been newer statistical ways to understand this, like for example, what is called as the time to even continual reassessment method, where during every cycle you actually look at what are the changes that happen from a toxicity profile and then bring it all together and say that, okay, what are some of the late onset toxicities that are being looked at. The second major deviation or divergence for immunotherapy agents with from traditional you could say, chemotherapy agents is the assumption which is called as the monotonicity assumption. What does it mean? It just means that when you actually give a cytotoxic chemotherapy, it goes and knocks off the malignant cells, and that is how you find out the DLT and the MTD, right? And the more the better was kind of like the mantra that was used. But for immuno-oncology agents, they don't actually impact the or impact the malignant cells directly. They instead act on other, you could say, immune cells like the T cells or the natural killer cells, and that leads to indirectly facilitating the cytotoxic efficacy of these drugs. So you need to now make a sea change from identifying the MTD to identifying what is called as the minimum effective dose. And to complicate matters, if you look at the dose efficacy curves, many of these drugs have plateau, which happens after a certain dose level. And sometimes the optimal dose level might even fall way below the tolerable dose or the dose which kind of like produces toxicity, etc. So if you actually say that we'll still stick with the DLT and the MTD, this could lead to finding incorrect dosing. Nivolumab, which was one of the drugs which were in the initial pathway, exactly had this issue. With the 3+3 design, they ultimately failed to even reach the MTD and then ultimately had to settle for a lower dose for development. And the second one is, say, for example, if you have, you could say drugs like anti-PD1 or PD-L1, etc., which are like immune checkpoint inhibitors and others, their relationship between toxicity and efficacy are also so different from lower dose to higher doses. So that could also be another thing which can actually happen. The third major divergence is what is called as the recommended phase 2 doses or recommendations for RP2 doses. What does this mean? So when a drug moves from phase 1 into phase 2, typically there has to be a recommendation as to what should be the phase 2 doses which are there. It's a critical step or a milestone in drug development. Now, implementation of, you could say, the DLT or some of these MTD, etc., in immunotherapy agents makes it very difficult in, you could say, identifying the recommended phase 2 dosing, because these drugs not only kind of like impact measures of efficacy like clinical response, but they also lead to alterations in pharmacokinetics, pharmacodynamic outcomes, as well as persistent immune response. So oftentimes with these kind of drugs, instead of the DLT and the MTD, the goal of the trial becomes identifying what is called as the optimal biologically active dose, or a concept called as OBD. And this kind of like has a lot of implications, because you're going to, you could say, incorporate both pharmacokinetic and pharmacokinetic data together in order to say what should be the, you could say, the maximum tolerated dose, etc., and which ultimately leads to your recommendation for the phase 2 dosing levels. And the last one that I would like to touch upon from the early development perspective is called as for the combination immunotherapy development. So many a time the combination drugs are now common for immunotherapy, whether it is with regards to using one drug, two drugs, or sometimes even three drugs. Three drugs are not very common, but two drugs are definitely the standard these days. And for them, you could say it's not very easy to find the MTD or the combination, because each drug would have very complex toxicity and efficacy profiles. And it is recommended that combinations of drugs with overlapping toxicity profiles has to be developed in order for them to actually make it, you could say, as to the best definition of DLT that is possible. So that's also another area where you can see that, yeah, you could say you need to have very different ways of how to identify, you could say, what is the maximum tolerated dose, etc. Now, this was for the early clinical development. Now, shifting to, you could say, some operational aspects of how safety is reported. Now, some preliminary, you could say, thoughts are that in immunology as well as in oncology drug development, whoever reaches the finish line faster makes all the difference for R&D investment as well as for the sales of the drug, etc. And early oncology development usually only involves a limited set of countries, right? It's like the countries with a faster turnaround time, readiness of the sites, ability to recruit large volume of patients, etc. These are very key, and with lot of data quality, right? And there is lot of, you could say, uh, uh, competition amongst countries to be part of these kind of drug development, and it's mainly driven by academic centers because some of the large academic centers which are there in the US, Australia, Korea, and in the western part of Europe actually very tightly control that they are able to manage all these studies, and it's very difficult for newer countries to actually join this. So we already have a lot of challenges with these, right? So for example, in India, the regulatory timelines, requirement for protocol amendments, capabilities, with all kind of like are stacked against us. And at the same time, and you'll hear about this in the next talk, is about how these changes with regards to how indigenous drug development and etc. will kind of like bring the value that India brings to the table. So when you have these kind of studies which are placed in India, there are significant challenges with regards to, you could say, some of these reporting of safety that becomes important. The first one is about investigator judgment, and we talk about this with regards to the safety perspective. If a patient in one of the studies had both existing chemotherapy and was on immunotherapy, the patient was on 4 drugs simultaneously. If the patient develops neutropenia, and if the investigator says that it is related, one of the big challenges is that how do you actually make the causality assessment that it is related to the study drug, right? So obviously, global organizations will try to push back, asking the investigator to provide more information. Many times, investigators, if they say that, okay, I don't want to discuss, or I'm not entertaining any of this discussion, that becomes a bit of a challenge with regards to, you could say, the whole thing. And some of the things that can be done is that the investigator can look at the line listings of the drug, the investigator brochure, and many of these things which can help in this. And one key thing is for the sponsor medical monitor to be consulted before the decision is made final. Another thing which is now important for, I would say, all the, you could say, pharmacovigilance centers as well as even for safety professionals is the activism that investigators and doctors now.
Sanish, sorry to interrupt. 2 minutes more, please.
Yes, yes. One of the big challenges with regards to safety reporting is the investigators on the social media, right? For example, we had a situation where the investigator actually put the patient details as well as, you could say, how the patient was on social media, and ask people to actually say, why don't you actually give us a recommendation as to what treatment to be made? And a bigger challenge is when the investigator says that, would you actually use a particular drug for treatment? And these are all representative. Please don't take these as the cases that are there. These are all representative. And what we all know that investigators do this in good faith, but sometimes inadvertently they disclose the name of the drug in a clinical trial, which can lead to a set of actions, which is patient activism, legal implications, sometimes even unblinding as well as regulatory scrutiny. So we say, investigators, you need to reach out responsibly, etc. There are also challenges with regards to, you could say, non-reporting of AEs, which usually come up in clinical trials in India. Sometimes it is because there are like the lack of understanding between a clinical trial and clinical practice. A classical example is nurses' notes, which might mention an adverse event, whereas it is not considered by the PI because it is dismissed as a common occurrence, etc. So these are some very unique things that we need to kind of like take care in India. So with that, I summarize, immunotherapy is now an established form of cancer treatment. They develop impressive clinical activity. And then, of course, because of the uniqueness of the safety reporting needs of these agents, both early clinical development and late clinical development has some changes that needs to be taken into act. With that, thank you once again, and happy to take any questions.
Thank you very much, Sanish, for that excellent presentation. You had brought out some exceedingly important point. I would request that we go ahead with Dr. Hasmukh Jain's presentation and take the questions of these two presentations together. Nitya, if that's okay? Yeah. So I'd like to introduce Dr. Hasmukh Jain, who is the Associate Professor Hematology and Oncology at the Tata Memorial Heart Center. And he's already very well known for his CAR-T work and has done a phase 1 study. And he has also set up several registries for hematological malignancies. So he's really going to speak from his own experience on the CAR-T safety. And he has already joined. So can I request Dr. Hasmukh Jain to please go ahead with his presentation?
Yes, ma'am.
Thank you.
I'm just trying to share the presentation. Okay.
You have 20 minutes for presentation, Hasmukh, and
Yes, ma'am.
then we'll have 10-minute discussion, yours and Sanish's together.
Yes.
Thank you.
So is my screen visible?
Yeah, Hasmukh, go ahead. All good. You're audible,
Thank you.
visible, all good. Yeah, please go
Thank
ahead.
you, ma'am. So thanks for inviting me. My topic is to discuss about the
Can you
safety of
make
CAR-T
this?
cell therapy.
Can you make your slides full screen, please?
Yeah, ma'am. So it is. I mean, to me, it is appearing in the full screen mode, but I'll again.
That happens sometimes. I don't know. I'm not seeing
I'll
it
just do
full
that again.
screen.
Yeah, I'll just. Okay, is it okay now?
Not really. I don't know about others. Can you all see it as full screen, Nitya?
We are not able to, madam, but maybe we should just go ahead in the interest of time.
Go ahead
Yeah,
then.
yeah. Please
All
go
right.
ahead.
Okay,
Thank
go
you.
ahead.
So I'll be talking about the safety of CAR-T cell therapy. So thanks for inviting me. These are my conflicts of interest. So CAR-T cell therapy is a form of immunotherapy. It's considered as a breakthrough innovation for relapsed refractory B-cell malignancies.
Your
So
slides are not moving, Hasmukh.
Hasmukh,
Sorry.
your slides are not moving, and I think you need to make it full screen for that, possibly.
okay.
That corner button, na? Yeah.
No, that I've done, but I don't think it's helping.
Okay.
Because to me it appears full screen. But what I'll try to do is in this. Is this better? Like
This is better. This is better, definitely. Yeah.
probably I'll try like this. So, CAR-T cell therapy is a form of immunotherapy. It's considered as a breakthrough innovation for B-cell malignancies. Now, essentially, what is CAR-T cell therapy is nothing but a modified T cell where we introduce the chimeric receptor. We make the cell to express a chimeric receptor against an antigen of our choice. So, you select a target antigen and then you make the cell to express that particular antibody directed against that particular antigen and use this for therapy. The unique feature is that you're combining the good qualities of a T cell as well as a B cell, one, and secondly, you're making the T cell much more potent. So it tends to proliferate much more rapidly, much more vigorously as compared to the normal T cell. So you can see that in the cartoon here, the single chain antibody fragment is the core component of the CAR T cell therapy, and it is linked to an intracellular domain, which is essentially important for the co-stimulation and for the potent proliferation of the T cell. It is a little cumbersome process as compared to the conventional chemotherapies and immunotherapies that we do. So what we need to do is, since this is a uniquely personalized therapy, for each patient we need to do the whole manufacturing process over and over again. So first thing that we do is we do a leukapheresis. So a WBC rich product is harvested. This product is then shipped out to a manufacturing facility where the gene editing happens. So essentially, the first step is that there is a selective T cell isolation by various techniques. Once we do the T cell isolation, there is activation of the T cell, then the introduction of the gene of interest. And there are different vectors that are used to introduce the gene. So this is either lentivirus or the retrovirus that contains the gene of interest, which transduces into the T cells and leading to the expression of CAR T cells. And we allow then for the subsequent expansion of the CAR T cell till we reach the desired dose. and only after doing a thorough testing for safety, identity, potency, and purity is the product either released back to the bedside or it is cryopreserved. Whenever the patient is ready, we then do conditioning chemotherapy, usually with a fludarabine cyclophosphamide-based regimen, followed by the infusion and then subsequent clinical management. So this is how the process usually goes. Now, the therapy has been so successful that there have been at least 6 products starting from 2017, which have got approved for various B-cell malignancies, ranging from leukemia to lymphomas to myelomas. The latest addition to this is these 2 products, where they've been approved for the use in myeloma. So there is tremendous potential, and it is also translated in the clinics. Uniquely, coming specifically to the topic of our discussion, is that it has certain unique toxicities. CAR T-cell therapy has certain chemotherapy related or conditioning therapy related toxicities like myelosuppression and infection, which we see with all the other chemotherapies as well. Whereas something that is unique to CAR T-cell therapy itself are these toxicities. So, I mean, in terms of frequency, the commonest one would be cytokine release syndrome, neurotoxicity. The third important thing and very rare toxicity, in which is of particular interest, is a possible insertional oncogenesis. So, CAR T cells, the whole process of genetic engineering can itself induce a malignancy. That's a concern. Another important thing is what is known as an on-target off-tumor toxicity. Depending upon what antigen is being targeted, there will always be some amount of expression of that antigen even on the normal cell. So how much ever we want, we will not be able to eliminate that possibility. And for CD19 CAR T cell therapy, which is the one that is the most active and most widely used in the clinics, other than the malignant B cells, it is also expressed on the normal B cells. So, and undesirable side effect of such therapies is B cell aplasia, which leads to hypergammaglobulinemia and therefore makes the patient susceptible to infections. The product itself, because it's a chimeric product, usually most of the times, it can lead to an anaphylaxis during the infusion. So these are the common and unique side effects of CAR T cell therapy, and I'll just take one by one, briefly discuss each of one of them. The most important thing is that this toxicity, to a certain extent, depends upon a host range of, a whole range of factors. Now here, it depends upon the therapy itself. It also depends upon the disease that you're treating. It also depends upon the patient population. So, for example, CAR T-cell therapy toxicity tends to be higher in the patients who have acute lymphoblastic leukemia as opposed to lymphomas. Similarly, if there is a higher tumor burden, that means that there is a higher antigenic load, and therefore a higher CAR T-cell activation will again lead to increased toxicities. Previous therapy, so if the patient does not have a good healthy marrow, again they tend to have a higher risk of toxicities. The dose of the CAR T cell therapy itself and the design of CAR T cell therapy again can influence these kind of results, which we have also seen in the clinics, and I will show in a summary table subsequently. Coming to the first and the most important side effect is the cytokine release syndrome. Now, you'll be familiar because this term was kind of very widely used in the context of COVID pandemic, where even COVID was known to cause cytokine release syndrome. This is something similar. So what happens is that this excess proliferation of CAR T cell leads to a hyperinflammatory response because of a vigorous activation of the immune system, and there's a release of various cytokines which manifests in forms of this in the form of this syndrome, which essentially has fever, hypertension, hypoxia as the key manifestations, but can involve any organ. So it can lead to cardiac dysfunction, renal impairment, hepatic failure, capillary leak syndrome. The severity ranges. So the mainstay of therapy is tocilizumab, and in advanced cases is systemic corticosteroids. I just show a summary checklist which summarizes how these patients are managed. So as you can see from serial point number 1 till 5 is how the severity of is when the severity of CRS increases. So if you just have an isolated fever, that generally falls into a grade 1 CRS when the blood pressure and saturation is normal, and most instances you just observe this patient and do symptomatic management. Whereas as the complications, as the symptoms worsen, that is the patient start developing hypertension, hypoxia, it becomes refractory, and this oxygen support or the ventilator support increases, your intensity of response also increases. So you start initiating tocilizumab. More importantly, the dose of steroids increase. So from Dexa 4 to 10 milligram, you go on to 10 to 20 milligrams 6 hourly, and in grade 4 CRS, you tend to use methylprednisolone 1 gram per day for 3 days, and try to kind of rapidly eliminate the CAR T cells from the circulation. So this is how generally cytokine release syndrome is managed. The second important toxicity, special toxicity of concern, is ICANS. ICANS basically is immune effector cell associated neurotoxicity syndrome. Now, this is a heterogeneous. I mean, it's a mixture of symptoms. So, starting from just subtle manifestation, like the patient, the earliest thing that they have is they have expressive aphasia. So, suddenly on the rounds, you find that the patient is not able to name objects. To finding difficulty in writing. So dysgraphia is one of the earliest manifestation, and then it can evolve to the extent that the patient could have seizures, and in advanced stages they could even have cerebral edema, and therefore could be or could go into altered sensorium, be delirious and comatose. So the exact mechanism of ICANS is not very clearly known. One of the theories is that it's probably because of the cytokines, but there's also a theory that it's because of the migration of the CAR T cells into the CSF. But this is something which is not settled yet. However, again, the management of neurotoxicity is depends on the grade, and here again I've summarized the way the neurotoxicity is graded and it is managed. So if we look at a combination of factors, we look at something called as a IEC score. Now, this basically looks at it's basically a simplified version of the mini mental status examination score, which which assesses the patient on 5 parameters, and you do a scoring. So if your score is 10, that is normal. But as the IC score starts dipping, that means that there is involvement of the higher mental function. You start treating the patient. So the grade 1 neurotoxicity where the patient is awake but has just initial subtle symptoms is where you start initiating DEXA. And unlike in CRS, for neurotoxicity, the mainstay of therapy is dexamethasone. And as progressively the symptoms worsen or seizures start appearing or the patient becomes comatose, the dose as well as the frequency of steroids increase. In addition, other supportive management, including anti-epileptic drugs and taking care of the ventilation, etc., is continued. And so this is how neurotoxicity is managed. Now, the third important problem that we face is B-cell aplasia. As I mentioned, this is one of the on-target off-tumor toxicity of CAR T-cell therapies, is something which cannot be completely avoided, and this leads to hypogammaglobulinemia. And both the products that are used by and large in the clinics, that is the anti-CD19 as well as the anti-CD22 CAR T-cells, can lead to these side effects. And as a result of which, patients can develop respiratory sinusal infections, and they generally require IVIG replacement. Fortunately, however, in most of the leukemias and lymphoma trials, it has been seen that in majority of the patients, there is a good amount of B-cell recovery over a period of time, and therefore these patients do not require lifelong immunoglobulin replacement. There are certain delayed toxicities. One of the important delayed toxicity is cytopenias. So usually chemotherapy leads to a transient cytopenia, whereas what is uniquely seen with CAR T-cell therapy is beyond the day 21, beyond the 3rd and 4th week, some of these patients develop grade 3, grade 4 cytopenias. In fact, with the commercially approved products, this was seen in up to 1/3 of the patients. And these cytopenias, again, the exact mechanism is not known, but these cytopenias could be long-lasting. So some of these patients eventually require a stem cell transplant to actually overcome these kind of toxicities. But the major concern also is that because of these persistent cytopenias, patients can develop bacterial and or fungal infections. So with the commercially approved products, this is one of the important concerns in the clinics. So if you look at the common toxicities and from the pivotal trials with the approved products, you'll appreciate that cytokine release syndrome, any grade of cytokine release syndrome was much higher, particularly with the murine products, that is Axicel and the Tisacel. And it remains so even with the other products. But the important thing is the severity. That is a grade 3, grade 4 CRS, that the incidence of this particular toxicity has down. Which is humanist CAR T-cell therapy, again, this toxicity has come down, which is what we seen experience, and I'll share those results as well. The second toxicity that we see is neurotoxicity, again seen in up to 40% of patients on an average of any grade, whereas the grade 3, grade 4 toxicity has the reporting has been very variable, so it ranges from anywhere from 2 to 20%. The cytopenias, as I said, is seen in about 1/3 of the patients. Now, these are the known toxicities for which we are now prepared, but there's certain unexpected, unusual toxicities also which have been reported. And one particular thing I would like to draw your attention to is something known as CAR T-cell induced lymphoma. So this is a unique thing that was reported recently. because this CAR T cell therapy manufacturing is a complicated process, and every time you have to do it from a patient's product, there have been attempts to use something called as allogenic CAR T cells. So you take the cells or the product from a healthy donor, and you then do the genetic engineering on those products and then use them for the therapy. So they would work like an off-the-shelf therapies, and you don't have to do the manufacturing process every time. So you could create those products and keep them ready, and then deliver them as and when needed. So a step in that direction was using something called as a piggyback modified CAR T cells. This is like a non-virus based approach. So viral vector was not used here for the first time, and they used something called as a transposon technology to develop the CAR T cells. What they reported the initial safety results of the first 10 patients. What they showed was that out of the first 10 patients, 2 patients developed a lymphoma, which on the biopsy subsequently was shown to be derived from the CAR T cells. So this is something that we need to be wary of, and because the experience is still very early, as we have patients who are getting cured with CAR T cell therapy, and as we keep them on follow-up, we will need to remain vigilant and look out for these kind of complications. And other concern is like neurological complications and autoimmune disorders which can possibly appear. So we need to look out for this. I'll just summarize briefly our own experience with our phase 1 clinical trial that we conducted. This was a part of a multi-center collaboration between national as well as international centers. The international centers, that is from NIH, they served as a knowledge partner. So we treated 10 patients of relapsed refractory DLBCL. So we used a humanized anti-CD19 CAR T cell therapy, and we gave a wide range of dose, starting from 10 to the power of 7 till 5 times 10 to the power of 9 CAR T cells, because unlike a general chemotherapy drug, you cannot be sure of the dose every time. So we allowed for a range, and also because the product that is infused does not always correlate with the in vivo expansion. So we allowed for a range because activity is reported over a wide range, and we gave it in a fractionated fashion. So split dose regimen over a period of 3 days, and so we concluded the trial recently, and we have we've had first 3 patients who got a dose of less than 5 million cells per kg, and then 7 patients who got a dose of more than 5 million cells per kg. What we noted was that cytokine release syndrome was manageable. We did not have a grade 3, grade 4 cytokine release syndrome, and neurotoxicity was also only grade 2 seen in 1 patient, but we did not have any grade 3, grade 4 neurotoxicity, and there were other toxicities which were generally manageable. So we did not have any therapy-related death. This is a comparison of the cytokine release syndrome and neurotoxicity with the other available products. And as you can see, with the humanized CAR T cell therapy, be it from the UPenn group or from our group, both the risk of neurotoxicity and the cytokine release syndrome is much less, particularly the more severe forms. Whereas with the humanized products with the murine products that are commercially available, these toxicities are a big concern, which again adds to not only to the cost, but also adds to the morbidity to the patient. So I would like to conclude saying that anti-CD19 CAR T cell therapy is safe and highly effective in relapsed refractory B-cell malignancies. There's a paradigm shift. We need to be aware of these kind of toxicities and be trained in how to manage these toxicities. We need to be vigilant for toxicities, unknown toxicities, which we may discover in the long run. So currently, the management strategies are essentially you have to do careful monitoring, recognize the toxicities early, and then prompt intervention. That is the mainstay. There are specific agents available in certain instances like tocilizumab for CRS and corticosteroids for neurotoxicity. These are my references, and I'd like to conclude. Thanks.
Thank you very much, Dr. Hasmukh, for that presentation, which was on a completely advancing areas, and also in terms of the various different dimensions and mechanisms of adverse reaction that we see in this. I now request all the participants who are, or the audience who are here, for their questions to both Sanish as well as to Hasmukh. And I'm not sure whether I'll be able to see your hand raised. Yeah, I can see Nitya has raised the hand. So I'd request her to please go ahead. And if others raise their hand, that'll be nice. And if I can't see Nitya, I'd request you to also see it. Sometimes, it happens. Nitya, please go ahead.
Yeah, thank you, madam. This is a question to both the speakers, actually. Sanish, if I heard you right, you said that the immunotherapy trials do not come to India, right? That's one. And yet you have Hasmukh presenting a phase 1 trial in the country. So what I wanted to ask you is, how do you as the industry or you as the investigator bridge this gap in drug development in the country? So maybe both of you could take it. Yeah, I'll just get the ball rolling and then we'll have the other people from the audience. Yeah. Over.
Yeah, so we'll have Sanish first and then Hasmukh.
Yeah, so while you say that, okay, yeah, there are challenges with regards to getting these kind of studies, especially the early part of the studies. I mean, the late studies are still slowly trickling in. I guess one of the big changes that will happen is that if we are able to showcase the kind of work that centers like TMH is doing or TMC is doing, etc., in this area, that would actually show that investigators in India are able to understand how to run these studies, then that will send a good signal to global organizations saying that yes, India is developing capabilities. We have the investigators as well as the infrastructure to actually manage that. And then I guess slowly, I think our regulatory agency will also kind of start supporting us from a timeline perspective and other stuff.
Yeah.
Thank you, Sanish. Dr. Hasmukh.
Yeah, ma'am. So I think one of the reasons why we went into developing cellular therapy in collaboration with IIT Bombay was because most of the immunotherapy products don't come to India right at the beginning. This is something which is unique, where you don't have to do like, it's not like a drug which is going to be controlled by a pharmaceutical company in that sense and will be marketed. This is something which is unique, where you can personalize and you can develop the capabilities indigenously, and then you can develop those therapies. So that is one of the reasons why we got into this. But I can understand from an industry perspective also why they don't get into this. Just from our experience, that the whole regulatory process is so complicated. I mean, given that this is cellular and gene therapy and this is first time, but even otherwise, the whole process to get the regulatory clearances took us at least 2 years before we could come out of the phase 1. And now with at least some experience, even from the regulatory side, for the phase 2 trials, at least we got it in 8 months. But I think still for a pharmaceutical company trying to come into India with these trials, these are like deadlines which are, I believe, will really make those critical decisions on whether they should get into India or not. So I think that is something where I think over a period of time, even the regulatory agencies will kind of get used to this and probably it'll get better.
Thank you, Hasmukh. I can see Dr. Arun Bhatt. Yeah, welcome, Dr. Bhatt. Yeah, please go ahead with your question. You're muted. You're muted, Arun. Just unmute. Need to unmute, Dr. Arun. No, we still can't hear you. Please unmute.
Okay. Yeah, now, it was supposed to be done by the organizers. I think just to support what Sanish said, no, I had recent experience that, the problem is with the regulatory guidelines are still in 19th century, while the phase 1 oncology and immunotherapy is in 21st century. So they still ask for the typical toxicity in 2 species and whatnot, which a startup doesn't do. The startup does a lot of PD work, good PK work, relevant toxicity, and goes to phase 1. So FDA will do it. They approve in 30 days. But here, I think we are back to mouse and rat being more important than immunotherapy. That's a problem.
Any comment from Sanish or Hasmukh on this?
Yeah, so again, the regulatory guidance or the regulations, the NDCT rules and others is all very agnostic of whether it is chemotherapy, small drugs, or large drugs or large molecules. So that is where the issue is, that the reviewers actually look at this without that particular distinction that immunotherapy or these newer agents need to be considered differently. So the data package that the regulatory agency expects is the same as that of a small molecule, which means that you do all these studies and then come to them. Whereas I don't think the patients can wait till then, right? I mean, we can't have another several years before we actually bring these drugs to the market. I mean, so that's, I think, where the change has to happen. And probably with the good work that institutes in India are doing, I think at least we will see some chipping away of these huge timelines or these lengthy timelines and it becoming much more competitive.
Thank you. Thank you, Sanish. And Hasmukh, any comment from your side?
Yeah, I agree, ma'am. So even with CAR T cell therapy, for example, there is globally there's no requirement actually to do studies on the mouse model, because it has been proven that mouse models do not replicate or it's not a good model to test for the toxicity. So anyway, you will not derive much information. But since it is there in the regulatory guidelines, they still insist. But again, as I said, so now once you start doing these trials and once you start going to the regulatory authorities, I think even for them it's a learning curve. So they now kind of become more receptive. Even our own experience has been that they become more receptive. So I think it will take time. We'll have to keep on trying.
Thank you. And also, I think the industry perspective, and Sanish mentioned that it's important to showcase the work being done in India so that there is more confidence internationally. And the government is also already working on setting up, say, more phase 1 units. So hopefully we will have some way forward on all that. I don't see any other hands up. I don't know, Nitya, can you see any more hands up? And I can't really see the chat box somehow. You know, in WebEx, it's a bit difficult. I'm not sure. Is there any question in the chat, Nitya?
Nothing that I can see. No.
So shall we close then this session?
Yes, ma'am. Yeah.
Maybe slightly behind schedule, but more or less caught up. And thank you
Yeah,
both,
yeah.
Anish and Asmukh, for this very excellent presentation. Completely new dimension to toxicities and also assessing them. And also over a period of time, I think maybe even the way we do the causality assessment will have to incorporate these observations. And at present, we just use the word relatedness and don't really go into the full WHO causality assessment especially when you're doing clinical trials. But this is a huge way forward and completely different way of looking at toxicity, as has been already mentioned by everybody. So I thank both of you and Nitya. I think it's this session. One more speaker is part of this session, isn't it, Nitya? Ocular toxicity. Is it this session or another next session?
Dr. Bhatt will take over now. Yeah.
Okay, great. Okay, wonderful. Thank you so much. Thank you very much. So over to Dr. Bhatt then.
Thank you, ma'am. We hope the first session has been a learning for all the attendees. We move ahead to the second session for the day. I invite Dr. Arun Bhatt sir as the chair of the session. Sir is currently consultant clinical research and drug development and holds the position of editor-in-chief of the journal Perspectives in Clinical Research. Dr. Bhatt has extensive experience of over 3 decades in the Indian pharmaceutical industry in clinical research, drug development, and regulatory affairs. His past positions held include President, Clinvent Research Private Limited and Medical Director of Novartis India Limited. Sir was formerly the President of Indian Society of Clinical Research and also the recipient of Drug Information Association Outstanding Service Award 2012 for his immense contributions in the field of specialization. I request you, sir, to kindly introduce the speakers and take the session forward.
Good afternoon. I think thank you, Dr. Nitya and the colleagues from KEM for this very interesting PVG session post lunch on a Friday. And I think it's I like the topics because they are not the standard run-of-the-mill topics about causality assessment. So that way, it's a great achievement. And I had a chance to hear Hasmukh and Sanish, and obviously it's a feast for the students and participants. So let me now start with the first speaker, Dr. Sachin Dharwadkar. Is currently holding the position of Director and Head of Glaucoma Services at Aditya Jyot Eye Hospital. Over 18 years of experience in the field of ophthalmology, recipient of ICO International Fellowship in Glaucoma, well known for research on modulation of wound healing in glaucoma surgery and ophthalmic imaging. He has published articles on automated perimetry, OCT in glaucoma and corneal topography in peer-reviewed journals, apart from chapters in books. I think the topic which he has chosen, ocular pharmacovigilance, I think is a very interesting one because as a physician and as a pharmacologist now, I understand that the eye is a kind of window to the world of the whole body. And now when we talk about eye, it's a kind of conglomeration of drugs, devices, surgery, medicine. And I'm sure Dr. Sachin will enlighten us about what are the challenges and what could be done about this special field. Dr. Sachin?
Am I audible?
Yeah, Sachin, we can hear you.
Yes,
Yeah. So first of all, thank you, Dr. Nitya, for having me here. Being in the August galaxy of pharmacologists and I'll try my best to I mean serve the purpose that I was made to be here for. So I'll just try to share my presentation.
Just go to the bottom. There is a share button.
Just a minute.
Yeah.
It's I think not allowing me to share.
1 minute. Tushar, please make Dr. Sachin co-host. Palvi and Tushar, you need to make Dr. Sachin co-host, please, so
Ma'am,
that he
sir
can
is
share.
co-host already.
Yeah. Sachin, just go to
I think it was
your co-host. Yeah, just try and share.
just a minute. I'll just join the meeting again
No,
because
you're
it's
co-host now. You should be able to share.
just a minute.
This WebEx now platform, including me, many people find it a little difficult. It's not something we do regularly. Just go to the share button, right corner.
So actually the Mac is not letting me share. That's the problem.
There's a share at the bottom, I think.
Hi, Sunita.
No, there is share at the bottom, but I think in
If you click,
system.
please click what happens. Just see what happens when you click it, because it will ask you to probably come back to your window and share the presentation.
Sunita, you're saying something. Could you type it in the chat box? It's not clear. Yeah. Sunita also has a Mac. I think she's trying to guide you.
It's saying open system preferences. I've already done that. I've allowed Cisco WebEx meetings to. I don't know what is happening.
Sunita, 1 minute. Tushar and Palvi, you need to unmute Sunita. She has a Mac, so she'll be able to guide Dr. Sachin.
Otherwise, Nitya, if you have a copy of slides, maybe you can present and Dr. Sachin can.
I think I've sent you.
Yeah,
Yeah,
hi, Dr. Sachin. I can see the problem is you will have to download WebEx and then
It is already downloaded.
then you have to go to the system preferences and give
Yeah.
permission to Mac to share the screen. I was facing the same thing, so I think that is what I did.
Permission to share. Where do you go to privacy firewall? Where do you go?
Go to system preferences.
I've gone there.
Just a minute, and I'll also try and download the slides till then. 1 minute, hold on.
Yeah.
So Tushar here, sir, for the technical help. So please click on that preferences in the back.
Screen sharing. Just a minute. Screen recording is there. Speech recognition, microphone, camera.
So there will be in the privacy option. Click on the Cisco WebEx and allow the permissions.
Private. I've clicked on the Cisco WebEx. And then what to do?
So you will have, I think you will have to exit and rejoin. I think that is how.
Exactly. That's what it told me. Just a minute.
Please try this, sir.
Yeah, I'll just exit and rejoin. Now it is getting shared.
Yes, we can.
Is
we
it
can see
seen?
it. Yeah, we can see it.
Yeah. Sachin, it's good. Yeah.
You can see it, right?
Yeah. Try and put it on full screen.
What I'll try to do is, is it seen?
Yes.
All good.
Yeah,
All
so
good.
I'll
Please
try
start.
to cover this in the next few minutes, in the next 15 minutes.
Okay.
So side effects and adverse drug reactions, I need not tell all of you, but that was for my understanding when I started thinking about this topic. We know these two are almost a continuum, and adverse drug reactions sometimes are slightly different than side effects, or sometimes are the extended version of side effects. So the problem for us as clinicians is always to understand the difference, because some of the side effects are the pharmacological extensions of the whatever the anticholinergic or anti-adrenergic properties of a drug. So whether to label that as an adverse reaction is usually a big problem, and that's why maybe there is an underreporting of side effects in ophthalmology, and essentially more because they are non-life threatening events, unlike the other faculties of medicine. So this is what a typical product drug insert looks like. And the problem is, unless you know what is the significance of these drug interactions clinically, it's very difficult to separate the news from the noise. So we cannot actually identify what is of clinical significance unless we experience it in clinical practice, because they give practically everything under the sun. So you can't usually isolate them, and unless they are of importance, it's very difficult for us to judge what is the most important of them all, or what is side threatening. So ADR for me is reactions that are significantly affecting the quality of life and significantly worse than side effects. That's the way I would put it. Ophthalmic drugs with local adverse reactions. The mechanism is being direct contact and absorption on the periocular skin. Now, ophthalmic drugs with systemic adverse drug reactions, absorption through the nasolacrimal duct, oropharynx, and the GI tract. And systemic drugs with ophthalmic adverse drug reactions, obviously they come into the eye through the blood, the uvea, the choroid, it being the very vascular structure next to the liver, and reaching the aqueous and the target tissue. Anti-glaucoma medications, fortunately or unfortunately, me being a glaucoma specialist, I have a lot of experience with these. And there are others which are less trivial in terms of side effect profile. So essentially, we start with the corticosteroids with local adverse drug reactions. Now, this is a very harmful drug. Over the counter, it has caused a havoc in people who are treated by general practitioners who don't know the side effects of topical steroids. And these do lead to ocular hypertension and open angle glaucoma or steroid induced glaucoma in many people, and also posterior subcapsular cataract. So these are things definitely you cannot call as side effects. These are more than side effects. This can affect the quality of life and need special treatment. Diligent follow-up and triage in predisposed cases is very important, because even a single drop of the lowest potency steroid, even like loteprednol or fluorometholone, even though they are said to be soft steroids, can cause IOP spikes to the range of 45 to 50 when the normal intraocular pressure range is 10 to 20. So something which we have to be very careful about in clinical practice. Allergic contact dermatitis is another bugbear. This can be API related or preservative induced. Predominantly seen with glaucoma medications, as the duration of treatment is chronic. Over a long period of time, the toxicity can be cumulative, or that can be idiosyncratic also. It can come up suddenly one fine day without any good reason. Commonly seen with brimonidine, dorzolamide, parasympathomimetic drugs, timolol, or benzalkonium chloride, which is the preservative with all these drugs. So it's a really difficult task to isolate, the treatment being removal of the cause. We have to actually isolate them in clinical practice and remove. Most of them are reversible. If you can see, the profile here is like a mild with this excoriation and this fissuring of the skin, whereas here you get a cicatricial ectropion. So this is the significant adverse drug reaction, more than what you can call a side effect. So it ranges from mild irritation at the angles of the eye to ectropion, and it's reversible on stoppage by and large if it is done before the stage of cicatrization. Otherwise, you will need to indulge into surgical treatment. Prostaglandin associated periorbitopathy is basically related to use of prostaglandins in the eye. As you know, it is the most important drug for treatment of glaucoma, and it has this drug has an effect on the orbital adipocytes, highest with bimetoprost and lowest with latanoprost in the spectrum. And these changes are very infrequently talked about, but absolutely cosmetically disfiguring consequences, leading to deepening of upper eyelid sulcus and enophthalmos. If you have a monocular treatment, especially in young people, it can look very ghastly, and definitely has to be considered as an adverse drug. Now, while describing this side effect, or rather this adverse reaction, it's not given due importance in literature. So as we have found out over the course of the last 25 years of using prostaglandin, this is a very significant issue, causing very tight eyelids, and sometimes it is reversible on stoppage of medication, and it may take over even weeks or months, and may also not lead to a complete resolution. So this is something which is very important and understated most of the time. Other than orbitopathy, we have issues like layering to be from some other person rather than the same person. If you can see, the asymmetry is very gross, and it can be also cosmetically disfiguring, although this can be usually reversible on stoppage of treatment. Periocular pigmentation and iris pigmentation are another issue. Iris pigmentation usually is irreversible as compared to the other pigmentations in the periocular region. Meibomian gland dysfunction also due to if affection of lipid metabolism in the meibomian glands, and this can cause dry eye. Now, dry eye can be caused de novo by these drugs, or this can be worsening of pre-existing disease. And the amount of morbidity that is induced by dry eye is very, very significant, and there is an extreme change in the quality of life of patient, especially those with glaucoma. And if they have pre-existing dry eye, these drugs significantly worsen the situation. Hyperemia, this is described maybe in all drops that the eye can become red, but hyperemia is not as simple as redness. Now, if you see, these eyes are red in a very, very different manner. This is absolutely tomato red, and this is slightly red with only exaggerated blood vessels. So, it can cause varying degree of ocular inflammation, episcleral vessel dilatation, and follicular conjunctivitis. Now, this over a period of time, if untreated and unrelenting, can go into ocular surface disease, dry eyes, and ocular surface failure. So, this hyperemia of this kind, you have to observe very closely. It's common with prostaglandin analogs. The one on the right side, this is the prostaglandin analog one, and this is common with brimonidine, that is red eye with follicular conjunctivitis, and especially 0.2%. What we have found that reducing the concentration to 0.1% has kept the clinical efficacy intact and significantly reduced the side effect profile. Now, these are also reversible on stoppage of medication, but what we have observed is, as a result of these medications, ophthalmic surgery, that is glaucoma surgery, has got affected a lot. Now, if you want to actually see technical definition of adverse drug reaction, this is the one, that is bilateral acute depigmentation of iris mimicking glaucoma and uveitis in fluoroquinolones. Now, this has been reported in like an adverse event actually in journals. This has not been a part of your routine ophthalmic literature that accompanies any drug. So this is seen with ciprofloxacin and gatifloxacin oral as well as drops. And this episode of acute depigmentation, it can definitely mimic a severe intraocular inflammation. So unless you are aware of this adverse drug reaction, definitely you will miss it and mistreat it for some bilateral panuveitis. Crystalline keratopathy and chronic use of topical fluoroquinolones also is a dose-dependent condition, and since we don't use antibiotics for a very long time, except in cases of corneal ulcers or post-transplants, we don't see it commonly, but you get these kind of deposits, and definitely these deposits are very, very difficult to get rid of unless we stop the drug and wait for a very long time. So this is also a significant adverse drug reaction of fluoroquinolones in the eye. Now, systemic side effects of ocular drugs, the basis, as we all know, is beautifully described in this chart, that the ocular absorption is only 10% of the dose that we instill, whereas the systemic absorption through the nasolacrimal duct and via the conjunctiva and the NLD is more than 50% of the dose. Now, what happens is, the bypass of first pass metabolism and direct systemic absorption causes, predisposes the patient more to the systemic effects, and there is no body weight basis for eye drops. So maybe these two things predispose patient to systemic side effects of drugs. Beta blockers, even a single drop has caused asthma attacks and heart blocks in predisposed people, especially those who are already on systemic beta blockers. So we have to be extremely careful as glaucoma surgeons to be aware of these side effects. Alpha agonists cause significant drowsiness to the tune of patient falling asleep for the next 2 hours after instillation of brimonidine tartrate eye drops. Sympathomimetic drugs, 10% phenylephrine has the tendency to cause arrhythmias and tachycardia. Although we use it less in clinical practice now, we use the 5% concentration more, but this is available in the market, and this has to be used with extreme discretion because this can be life-threatening. Carbonic anhydrase inhibitors, used orally as well as topically in our case, and this can lead to loss of appetite, and interference in taste is the most important adverse reaction that we have found. And obviously metabolic acidosis and hypokalemia are very common, although we give potassium supplementation, but the adverse reaction is very variable in patients. Some people become extremely morose and listless, and some of them can tolerate this drug very well. So there is a huge spectrum to it, and we have to be very, very vigilant about the side effects. Parasympathomimetics we use very infrequently, but we have seen patients undergoing bronchospasm and gastrointestinal motility problems with parasympathomimetic drugs, but to a much lesser extent. As far as antibiotics are concerned, sulfa-based antibiotics, as we know, have even caused Stevens-Johnson syndrome in predisposed individuals, which is definitely adverse drug reaction. So we are extremely careful if the patient is presented with a previous history of sulfa allergy. Although the use of sulfa moieties in acetazolamide does not happen to cross-react with these drugs, and we have been using that pretty safely, because as we all know, the structure of antibiotics and other sulfur derivatives may be completely differing in the side effect profile. Steroids, definitely we already discussed. Topical periocular cause hyperglycemia and endocrine consequences, but to a much lesser extent than what systemic drugs can cause to the eye. So that is something, that difference we should always remember. Now, strategies to minimize side effects, which we use as glaucoma specialists. Just for general information, I'm narrating: detailed prescription, proper advised and supervised installation method. Social support is very important. Eyelid closure post installation for 3 to 10 minutes. In letter and spirit is very important, because if you don't do it, then the nasolacrimal drug causes all the damage, and then your drug is into systemic circulation at 50% of the availability, and then you have problems. Elimination of skin spillover using tissue paper is also very important to prevent the absorption through the skin. And punctal occlusion for 3 minutes. We have all these mistakes that I've highlighted on the right hand side of the slide. And this is how the puncta are to be occluded, because the nasolacrimal anatomy is not known to the patient and not properly demonstrated by physicians. But this can definitely reduce the systemic adverse drug reactions by a mile. So this is something that is very important. Now, coming lastly, the systemic drugs with ophthalmic adverse drug reactions, they can be present in two major classes. It can be due to drug deposits or collateral effect on the autonomic nervous system. Now, this can be dose related or idiosyncratic. They affect the cornea, lens, retina, and the intraocular pressure. These are the four main areas where it affects the eye. The list of the drugs we have seen on the right hand side, I've already pasted, and these are in the order of importance that we see in clinical practice. And the drug inserts that are there with these medicines contain everything except the clinical significance. So that's why we'll discuss at least around 5-6 of them, which we see can affect the patient's life in a very drastic way. So as I already told you, corticosteroids have an effect on the trabecular meshwork morphology, polymerization of glycosaminoglycans, and expression of extracellular matrix, and they lead to severe intraocular pressure responses in predisposed individuals, 1/3 of the population to be specific, and they need to be aware. Otherwise, this intraocular pressure elevation, if it is slow, patient may not make out, and they will land into optic atrophy by the time they present to us. Posterior subcapsular cataracts, very common with the chronic use of steroids, especially for allergic conjunctivitis and all those indications, and also oral steroids for maybe say renal transplant or a bone marrow transplant. And a central serous chorioretinopathy, which involves, if you see this diagram, swelling of the retina, there's fluid accumulation, where steroids in any form, maybe topical, systemic, or whatever, are supposed to be avoided to prevent the recurrence of such a condition. Now, another most important bugbear is basically anti-tubercular drugs, especially ethambutol. Now, optic neuritis and central vision loss with central scotoma is very common due to ethambutol, especially if it exceeds more than 35 milligram per kilogram per day. The problem is that when the patient is suffering from COX, he has a sudden weight loss, and if the dose that you give for ethambutol continues to be the same, then definitely these patients are predisposed to a sudden onset of optic neuritis, which can cause bilateral central scotomas and sudden decrease in vision. Similarly, isoniazid on prolonged usage does have an idiosyncratic effect, but it is less severe and reversible on stoppage. Linezolid also has caused, which is a second line AKT drug, has caused a few episodes of optic neuritis, more than 28 days of usage. So all in all, it suffices to say that all three drugs can cause, but ethambutol is the most important, and we have to be very vigilant as ophthalmologists and even as chest physicians about the side effect of ethambutol. Color vision and visual fields are very vital to the early identification, and we always, when patients come to us, we always tell them about the importance and what are the warning signs to identify in such a case, because ethambutol toxicity has to be identified early to prevent irreversible visual loss. As far as the chloroquine and HCQs are concerned, they are anti-malarials and DMARDs, as we know. But the DMARD is the problem, because anti-malarials are a short course, and this is a cumulative toxicity. So nowadays, with new equipment like spectral domain OCTs, we don't have to wait till the stage of bullseye maculopathy, but we can identify by multifocal ERGs or this kind of a what we call as a flying saucer sign on the spectral domain OCT. The moment we identify this, we have to stop the drug, and this will prevent the irreversible loss of photoreceptors and the ellipsoid layer in the central retina. So high index of suspicion counseling is mandatory, and we obviously the patients are referred to us every 6 months, say for a retina evaluation. Then we have to look at these very closely. Obviously, we don't wait for the stage of maculopathy, the one which I've shown you above. But definitely, we have the imaging techniques now to identify these ADRs early and then nip it in the bud. Another important of ophthalmic adverse drug reaction, basically you can call as an extended side effect, is of tamsulosin or alpha-1 blockers. It blocks the prostate smooth muscles as well as the iris at the same time. Now, what this leads to is that in each and every patient who has a tamsulosin, we have to be ready for a iris prolapse like this. You can see at the end, during the surgery or at the end, you can have iris popping out of all the incisions that you have made, makes the surgery a living hell. And in those cases, the pupil also is very small and floppy, so we have to use something like iris dilators to keep it in proper shape to complete the surgery uneventfully. And this is extremely common side effect, and we anticipate it in every patient of using urinary relaxants. It is managed with intraoperative intracapillary adrenergics, pupil dilators, and high viscosity viscoelastics. And we've learned over time now, because the frequency is so high, that we've almost mastered the management of this particular problem. Another dangerous complication is with anticonvulsants, topiramate and vigabatrin. Topiramate is very commonly known to cause an angle closure glaucoma because of supraciliary effusion. It's an idiosyncratic reaction again to the sulfa moiety, and it causes bilateral acute myopic shift with angle closure. Now, multiple cases have been reported. We've ourselves seen around 3 to 4 cases of this adverse reaction. So, and topiramate is a very commonly used drug by many people. So, it's important that this adverse reaction is known to everybody, and this drug especially has a very common usage in practice. So we have to be very careful. IOP lowering medications and cycloplegia do help us treat most of the cases very well, but in extreme cases, sometimes you have to resort to maybe admitting the patient and maybe giving IV mannitol or some, because the intraocular pressure spikes are extremely high and maybe go to 45 and 50 and cause ischemic damage to the optic nerve due to the acute elevation. Vigabatrin is another drug. The unique problem to vigabatrin is that it causes irreversible visual field constriction. Now, the problem is that this drug is used in pediatric age group. Patients may not be amenable to computerized visual field testing, and it occurs in 30 to 50% of all patients. That's a very, very high incidence. So we have to be very careful. And since it is irreversible field constriction, it's a permanent affection of quality of life. So care has to be taken about the dosage. We have to be very careful, and we have to be very vigilant about this adverse drug reaction of vigabatrin. Amongst the last few, amiodarone, the antiarrhythmic drug used very, very commonly. It causes vortex keratopathy in 98% patients who receive more than 200 to 300 milligrams per day. Although it just looks like a fancy star on the surface of the cornea, doesn't affect vision much. That's why I kept it almost at the last. But these are deposits with intracytoplasmic inclusions in the epithelial cells and can cause deposits in the other structures like conjunctival lens epithelium and conjunctival fibrocytes. But this by and large is usually asymptomatic and reversible. So this ADR can be that way a very trivial one as compared to what we discussed till now. Anti-neoplastic agents have their own set of problems. Most of them are basically related to the ocular surface, that is the lacrimal drainage pathway and conjunctivitis, and even blepharitis. And some of them are definitely related to the retina, that is cisplatin and nitrosurias, which can cause optic nerve atrophy and optic nerve toxicity, and also vincristine. So something that we have to remember, that the important ones are vincristine, cisplatin, and nitrosurias. With the adverse drug reactions, we have to monitor the optic nerve very, very carefully. Ophthalmic reference should be done, and we should be on board, because now we have instruments and devices that can identify all these problems early. As far as the ocular surface is concerned, much more trivial and much more reversible and amenable to treatment. It is the optic nerve and the retina that you have to be very careful about. So in summary, knowledge of ADRs and their scope can limit the associated morbidity in their deployment in patient care. Triaging and tiering of side effects in our knowledge base can prevent disastrous consequences in practice, often life and sight threatening. And regular updation of ADRs in PVPI and their dissemination can be very useful in clinical practice. Thank you for your patient hearing.
Thank you, Dr. Sachin, for a very interesting and I think a very wide scope of different issues in ocular pharmacovigilance. I think it's an eye opener, if I may say so. And maybe I will request now the audience to ask maybe 2 or 3 questions on this subject. I think Nitya has raised her hand.
First of all, Dr. Sachin, thank you. It was really an eye-opener, as Dr. Arun had said. Actually, there is a private question. It's a question which has been sent privately to me. I think we are focusing too much on reporting of ADRs, but not about preventing ADRs. So maybe you could tell us how you sort of work towards prevention in ophthalmology. You did touch upon it. You've talked about OCT and other things, but maybe you could add something. Over.
See, basically, once we know, see, most of these reactions, for example, like the allergic contact dermatitis, the cumulative toxicity of hydroxychloroquine or ethambutol, these are the ones which are the more dangerous ones, which are sight threatening. So the awareness about these ADRs is there. So what we do is it is already factored into our examination pro forma or examination schedule. So whenever a patient who's referred to us or whom we potentially know is on these medications, we always put them on alert. Like, we tell them the signs and symptoms of what is supposed to be done as far as the systemic medications are concerned, and the kind of scotomas they are supposed to anticipate. We have something like the Amsler's monitoring, home Amsler's monitoring for macular changes. We tell them about color saturation for optic nerve head problems. So all these things are always there. Intraocular pressure monitoring for patients on corticosteroids is mandatory. So in case patients are on topical or systemic corticosteroids, we always have a written schedule. Written schedule is very important, and not only in writing, but the patient has to be told. So communication for us glaucoma people is very, very important, and we extend that to all the other drugs because it comes as a second behavior to us, unfortunately. But the table time for us is very important, making them aware, and obviously we being aware of the adverse drug reactions is very important, especially for glaucoma medications. We have been doing this programs all over India. I am a faculty for the Glaucoma Society of India. So these ADRs of glaucoma medications are the most important in ophthalmology as far as ocular leading to systemic is concerned. So all other medications are very trivial in terms of systemic side effects. So we've been highlighting this at all meetings, and by and large the information, the what do you call, the transmission of information has been very good. So awareness is very, very important, and we, as I told you, the right method of punctal occlusion is a game changer. So it has saved many lives. The right method of drop installation, the right method of punctal occlusion is so very important. And let me tell you that 90% of the patients who are referred to me in my practice do not even close their eyes after putting the drop because the communication is very poor. So what we do as glaucoma specialists, or what I do, we have a very, very detailed discussion with the patient. We have a lot of table time that we give, and all these small things which I've told you today are told to the patient in no uncertain terms, because this can be life saving. We have seen bradycardia, syncopal attacks, patient going into heart blocks with a simple 1.5% timolol maleate instilled into the eye when the patient was on systemic propranolol. So these things are very, very important, and eye drops, though appear trivial, can be life-threatening. A single drop of timolol maleate also can cause an acute asthmatic attack. So we can prevent all these ADRs to a significant level by awareness and education, and obviously patient communication and strict protocols.
So Sachin, there are 2 questions. One is, of course, a question on your current, I think, explanation. Why punctal occlusion is done? Can you explain in brief?
Why?
Punctal occlusion is done. Can you explain why? That's a
So what happens is, because as I already showed you in the slide, the bioavailability of the drop in the systemic circulation, if you don't occlude, is around 50%, whereas on the eye it is 10%. So what happens is, if you don't occlude the punctum and if you immediately open the eye, the lacrimal pump will throw all the drug into the systemic circulation through the nasolacrimal duct. So it doesn't have to go through the liver. The nasal mucosa just takes it in, and it is directly available in systemic circulation. So to prevent that from happening, what you do is basically you occlude the lacrimal sac. Now the problem is the sac is located in the lacrimal fossa, which is pretty inaccessible. Like, this is the point, inside. So people, what they tell, they do it here, everywhere on the nasal bridge, but it is actually in the lacrimal fossa, which is pretty deep. So this is the point. The photo that I showed you, is not very easy. And even if patients have been told punctal occlusion, you have found that 99 out of 100 times they are pressing on the nasal bone. So this simple punctal occlusion for 3 minutes can prevent the drug being bioavailable in the systemic circulation in a higher dose that can cause side effects. So 3 minutes by our clinical judgment is fine. What I tell my patients that the eye closure should be for 10 minutes. All the books say 3 minutes. I tell them to close for 10 minutes, and I've seen that the pharmacological effect of the drug inside the eye, as far as the ocular hypotensive effect is concerned, is excellent, as well as the side effect profile is very minimum. So 10 minutes eye closure with 3 minutes punctal occlusion has saved many lives and many side effects. So this is something that we should all remember.
Dr. Shirsagar has a question. Madam?
Yes, I just wanted to add here to that very excellent presentation from Dr. Sachin that recently miltefosine, which is a anti-leishmanial drug, has also been shown to have ocular side effect. That's just one addition I wanted to. Thank you.
Any other question? Because I saw one Dr. Purohit showing me a hand, but I think is it just a passing hand or I don't know. But I don't see it in the box. So thank you, Dr. Sachin, and we'll go to the next presentation now from. Thank you very much.
Thank you so much, Dr. Nitya. Thank you. Thank you, Dr. Arun.
Thank you. So next we have an interesting talk by Dr. Sunita Shukla. Dr. Sunita Chapola Shukla is currently a visiting consultant in the Department of Pharmacology at KEM. She is MNCNT from Nair Hospital and went on to become advanced allergy training from Massachusetts General Hospital, MGH, Harvard, and Boston. She is the founder of Mumbai Allergy Center, and her work in COVID vaccine allergy testing contributed to improving the vaccine drive at KEM to the medical field as well as whole during the pandemic. So she is going to focus more on the issue of allergy as a issue and allergies in the drug safety. That seems to be her focus topic, and I think it's again a very interesting, diverse topic compared to the standard topic, and we'll look forward to Dr. Sunita's presentation. Dr. Sunita?
Yeah. Can you see my slides?
Sunita, your screen is visible, your computer laptop screen.
You'll have to select.
Yeah, one minute.
Yeah, you'll have to select the slides and now show them. Yeah.
1 second. One second. I'll just stop it and I'll restart.
Yeah. Take your time, Sunita. Because, sir, there is a comment in the chat box. Dr. Sachin has delivered an informative and excellent lecture. I think everybody agrees with that.
That part I thought was given, but so I didn't read out, but I think I saw the comment. But yes, that's true. Definitely.
Yeah, it's true. Absolutely an eye opener. Yeah.
Yes.
Sunita, all good. Yeah.
Yeah. Can you hear me?
Yeah, here,
yeah, okay.
yeah. All
we
good.
can see
Yeah.
you.
Please get started.
Yeah. Okay.
My camera is off, I guess.
Oh, we can see your slides. I think you
Okay.
probably need to start
Could I
your
put
video.
on my camera? I think I can't find where it is.
It's okay, Sunita. Maybe you can switch it on after you finish.
Okay, because this WebEx is with...
WebEx is like that only.
Yeah.
Okay, 1 second. I'll just... Yeah, it's on.
Yeah, we can see you now. Yes.
Yeah. Okay. Good afternoon, everyone. Thank you, Dr. Nitya ma'am, for giving me this opportunity and making me learn something about WebEx also. We are so used to Zoom. So today I will be talking about drug allergy, and as Dr. Bhatt said, post-lunch session drug allergy is definitely puts you to sleep. So I will try not to do that by putting some interesting clinical cases. So as we all know, drug allergy is an immunological mediated response to a pharmaceutical agent or formulation agent in a sensitized person. So as we have seen in the few lectures before, drug reaction are of two types. One is a type A and the type B reaction. So type A reaction is a dose dependent, predictable, and the most common reaction, and it is seen in more than 80% of the people. And this is usually due to overdose, side effects, or drug interaction. Whereas type B reaction is a dose independent, unpredictable, and very uncommon, and it is seen in less than 20% of the patient. And most of the time it is because of intolerance, idiosyncrasy, and drug allergies. Now, drug allergies are usually divided into two types. One is an immunological reaction and other is a non-immunological or a pseudo-allergic reaction. Immunological reaction we all have studied since our MBBS days. It's the Gell-Coombs classification, and we have 4 types. Today we will be talking more about the type 1 reaction, where which is actually a immediate reaction or a IgE mediated reaction. And what we usually see is anaphylaxis, urticaria, angioedema, bronchospasm, hypotension, and so on. And a type 4 reaction is a T cell mediated reaction, usually seen in the skin when topical agents are used. So there are various risk factors for having a drug allergy reaction. So the drug factors are the nature of the drug, the degree of exposure, the route of administration, cross sensitization with other drugs, and the various host factors which make a difference are the age and sex of the patient. It is commonly seen in females and males. The genetic factors, especially the HLA type, the concurrent medical illnesses, especially the virus, previous drug reactions, and whether the patient has multiple allergy syndrome. So the mechanism of drug allergy is many, but the commonest I will be putting here. We have the IgE mediated reaction. This is a mast cells and it has a FCE receptors. So when we have IgE antibodies on this receptor in an already sensitized person. So once an allergen comes into the bloodstream or the body and it attaches to these antibodies, and the mast cell degranulates, causing release of many mediators like histamine, prostaglandin, leukotrienes, and so on. So these mediators of hypersensitivity and inflammation causes various clinical symptoms like smooth muscle contraction, which we see in asthma. There is vasodilatation, secretion by mucus glands, and so on. There is another very common mechanism of drug allergy, that is the haptin protein complex. Here, a drug or its metabolite or a degradation product are usually non-allergenic. But once they combine with a similar protein in the body, which may be non-allergenic, they cause the formation of a potentially allergenic substance. And these are the hapten protein conjugates which cause all the reactions. The commonest drugs which we see clinically are the antibiotics, where the penicillin or the beta-lactam group are the commonest. We have Septran, aspirin, NSAIDs, chemotherapeutic drugs, anesthetic drugs, anticonvulsant, and so on. But the beta-lactam, aspirin, and sulfonamide make the commonest scenario, that is approximately 80%. So diagnosis of drug allergy in clinical scenario is done mostly by a good history taking. Here, the onset of the signs and symptoms and the temporal relation with the taking of the drug is the most important. On examination, usually the patient comes to us once he is better, but usually at the time of reaction, the primary physician sees this patient first, and depending on what the patient presents with, it could be a systemic reaction, like there could be palpitations, tachycardia, hypotension, or there can be in general rashes, urticarial rashes, or the wheals, and many times anaphylaxis picture also. So these things will be treated by the general physician or the primary physician of the patient, and then the patient is referred to an allergy specialist for the testing. The in vitro testing, which can be drug allergy, are the drug specific Ig levels, basophil activation test, and the serum tryptase level. Here, the drug specific Ig level are done, but they are not very sensitive. We get many false positive in that. And because of this, we should not label a patient as drug allergy for that substance. Serum tryptase level is good only for post anaphylaxis patient, and it should be done within 2 hours of anaphylaxis, and if it is raised as compared to the basal level, it is important. But the most important tests are the in vivo test, which are the skin prick test, the intradermal test, and a challenge test. So this is a brief detail about how do you do investigate a drug allergy. Once you suspect that the patient is an immediate Ig reaction, we go for the skin prick test, and then the final answer is with the drug challenge test. And if it seems like a delayed T cell mediated, then we go for something called as a patch testing, which is done on the skin. So skin prick test is a gold standard diagnostic method of diagnosing most of the IG mediated allergies throughout the world, and it is accepted by all the major allergy organization of the world. In skin prick test, we put a drop of the allergen, and with the help of a needle with the bevel up, we take a very modified prick where we just elevate it so that a drop of the allergen enters into the basal layer of the skin. Once this happens, if the patient is allergic to it, we get a wheal and flare reaction in 15 to 20 minutes. Similar to this, we have an intradermal test where with the help of a 26 gauge needle, we just inject the drug in the layers of the skin, and with the bevel up, and a very small wheal is raised as is seen in the picture. In drug challenge test, once the patient is negative to the skin prick test and the intradermal test, we go for the drug challenge test, and this is the gold standard for diagnosing and proving drug allergy. It can be a oral or a IV method, and it has multiple step approach. Here basically small amount of the drug is given at regular interval with proper monitoring of the systemic parameters till the time full dose is received. And if the patient doesn't react to it, the test is negative, and if the patient reacts to it, it is a positive drug allergy test. This is how a patch test is done. Many chemicals or drugs which are used topically or many other substance can be also put like this on the back, and the patch is removed after 40. So I have presented few interesting cases here which will make this very easy to understand. So this is a case of a young female who had a history of Norflox allergy in the past, and recently she also had a history of amoxiclav, and she developed a rash and wheals throughout the body which was decreased after using antihistaminics. Now she wanted to undergo a dental procedure, and the doctor wanted to give her Ciplox, because as we all know, in dental, Ciplox and Flagyl is the commonest used drug. So she was referred to me to know whether she still has a Ciplox allergy. Now this is the reaction of, we can see that there is a skin prick test, and there is a serial dilutions of IV Ciplox, and we started from 1:100. There was hardly any reaction, but when we went to 1:10, you can see there is a wheal with a flare. But when we went with the highest concentration of 1:1, there was a big wheal with a flare reaction, and this is a positive test for it. Now the second question was, if she is allergic to ciprofloxacin, what are the other antibiotics we can give for her during her dental treatment? So we discussed with the dentist, and we decided to go for cephalosporin, and that was ceftriaxone. And we did the testing for the same, and here you can see there is no reaction to either the skin prick test or the intradermal test. And after this, we give a challenge test of cefixime to the patient, which she tolerated very well, and later on she underwent the dental process without any problem. Similarly, this is another case of ciprofloxacin. This is a 52-year-old male with various other histories, and he had a reaction to Ciplox 20 years back, and Norflox 1 year back. So he was a very educated, high-class patient. He just wanted to know, does he still have Ciplox allergy? And this is the same test we did for him. We can see the skin prick test. There is a very small wheal of 3 millimeter, but the time we underwent for an intradermal test, as we increase the concentration with 1:1, we can see a big wheal of 2 by 1.5 centimeter with a pseudopodia. The more the pseudopodia it is during the skin prick test, the more severe are the allergy. And the flare you can see is more than 4.5 centimeter diameter. So this is a very severe reaction, and with this much amount of the amount of solution which we use for intradermal is only 0.02 ml to create this wheal. Now this amount gave rise to itching in the ear, and on the trunk you can see the rashes also. So God forbid, if this patient who has reacted to a skin prick test where he's getting a systemic reaction, if we give him a syrup or a tablet of Ciplox, chances of him getting an anaphylactic attack is very high. So he has to avoid it for as long as possible. So now he wanted to know alternative antibiotics so that he can use it if he has any infection. So we did a testing for amoxiclav, and here we can see that it is negative. These are the wheals which are created after injection, and he even tolerated the oral challenge very well with 625 milligram. This is a case of a young doctor with some allergy histories, but she had always taken Ciplox before in her life. But once she took Levoflox, she developed this rash and a bulla, which is a typical of a fixed drug eruption. And this is how it healed within a month. Now, after a year, she had some diarrhea and she took a dose of Ciplox TZ without realizing that she had a Levoflox reaction. And within 1 hour of taking the dose, she developed the same erythema at the same site where she had the bulla last year, and this is how it healed within a month with desquamation. So this is very typical of a fixed drug eruption, that it will happen at the same place every time, the itching, the bulla, the desquamation. So if we have to do testing in such FDE patient, it has to be done at the local site. If we do it on the forearm or the back, the chances are that we'll get a negative result and we'll say that she is not drug allergy to this Ciplox, but this is typical of FDE. This is a similar fixed drug eruption to ofloxacin. This is a patient we saw at KEM, and this is how the lesions will typically heal with hyperpigmentation. So we get lot of aspirin sensitivity also, and many times the cardiologist wants to start aspirin, but the patient doesn't tolerate it. If the patient gets rashes and all, then we can always do a desensitization of the patient. Here we start with small doses of the aspirin over a limited time, and we go on increasing the dose till the patient tolerates a full dose. So this desensitization is a very temporary process. If you stop the drug for anywhere between 2 to 5 days, it has to be done again. So this is only a temporary tolerance. This is another very interesting case of an old lady who was labeled as a lignocaine allergy 20 years back when she was undergoing a cataract surgery. And because of that mislabeling, she has never ever used lignocaine, nor the doctor has used it for her. But now when she went for a root canal treatment, she had to do it without the help of an anesthetist. Now, this is very scary that you are undergoing a RCT without the help of a lignocaine. And it was a nightmare for her, and she was referred to us, and we did this testing. And here you can see that the skin prick test, intradermal, everything is negative. In intradermal, we went for the full concentration of 1% and then 2%. And then for the challenge, because this is injected subcutaneously or into the mucosa, we did a subcutaneous testing with a 5 ml, and she tolerated it very well. And then next RCT she underwent under the cover of lignocaine. This is a very young boy of chronic renal disease, and he developed a Steven-Johnson syndrome after taking some penicillin drug. And he was awaiting his kidney and liver transplant. And before that, we actually did many of the drugs for him, whether he's allergic to it or not. And whatever we did, he was negative. He got transplanted, but unfortunately there were other issues, and this is how he reacted to the micropore, the dynaplast which was attached for the IV cannula and all. And this started a reaction. He underwent into sepsis, and we lost this young boy. And then retrospectively, on discussion with on various physician, we realized that every time he was undergoing a dialysis, they were using a dynaplast, and here this time they have used a micropore, and he reacted to the micropore. So this was a eye opener for us, that this micropore can itself cause contact dermatitis and peeling of the skin like this. So this is another case of contact dermatitis. This patient is from Nasik, and she had these lesions on the hand. And when we did a patch testing for her, this is the patch testing. You can see the lesions on the right, they were severely positive. And this was actually to copper and pitambari. And then we realized that they have the habit of sitting in the bathroom and washing utensils. That's why the typical lesions on the palm and the sole. So on discontinuing the copper utensils and the use of pitambari, they have started using steel, and now she is much better and all the lesions are completely healed. Similarly, this is a hand of a young boy, a sports person. He had this typical lesions on the hypothenar eminence and the fingertips. And when we did a testing, it was positive to the black rubber, which is present in the cricket handle bat and the tennis bat. So after discontinuing that use, he is much better now. This is a patient of multiple drug allergy. He was referred to us during COVID time. And here you can see this is a prescription of Kasturba Hospital. This many of us has faced that a patient is mislabeled for multiple drug allergy. Here we can see there are 3 antibiotics, NSAIDs, and even a PPI, rabiprazole. And it was just blanket rule. It was written vaccine cannot be given. So this boy, when he came to us, we at that time were doing a vaccine component test, that is the PEG and the PAT and all. But we did it, it was negative. But this patient then wanted to get tested for Ciplox. And we did his Ciplox testing. And if you can see, the red one is the reaction, the wheel, which was positive after injecting the maximum dose. So we asked him to refrain from ciprofloxacin, but he could take the other drugs carefully. So we know that in this era of COVID, there were a lot of issues regarding the vaccine. So the COVID vaccine or any other vaccine has usually 2 components. One is the biological agent and other are the excipients. These excipients in COVID vaccine are usually the PEG and the polysorbate. And these are the culprit who can cause a direct reaction. So initially when we started the testing, madam, and we had lot of talk, and we started testing these people for PEG and PAT. Because PAT is cross reactive with PEG, and PEG and PAT are very common substance in our daily living, like they are present in our cosmetics and even in the toothpaste. So if a patient was negative for this, we asked them to undergo for a COVID vaccine. And this was the paper which we released from KEM regarding the evaluation of such people and the allergic reaction, whether they had it or not after COVID vaccine. This has a free online access on JAPI. And then came the idea when madam lost one of her colleague to COVID, and we realized that there shouldn't be a blanket rule by the government that if a patient has allergies, don't give the vaccine to it. So with this, there was the birth of this prospective study where we tested COVID vaccine along with PEG and PAT at KEM. We successfully did it for 100 patients. And this was only one patient where we found a reaction to the undiluted dose of COVID vaccine. But later on, the patient successfully took the COVID dose also. These are the unusual cases of drug allergy. Here, this patient, I was doing a COVID vaccine test, and I was realizing that for every dilution, she was getting the same wheel and flare reaction. And then I realized this is not possible, because as we increase the concentration, the size of wheel should also increase. Since it was not happening, then we realized that she is reacting to saline also. And on questioning to her, she said that whenever she has taken saline in a hospital, she always used to get rigors. So maybe this is a hyper reaction to a normal saline. This is a patient who was positive to cetirizine. I couldn't believe it, but when she gave a very typical history, which had a very good temporal relation, we gave her 1/10 dose of cetirizine, because we don't have the IV form, and we couldn't do the skin prick test and the intradermal test. On giving her the 1/10 dose orally, she immediately within 15 minutes started developing the rashes. This is a very rare case of AVIL or the chlorpheniramine Marriott allergy. This is a ICU sister of Global Hospital, and she was admitted during COVID, and she, after having some rashes, she was giving AVIL for that, and she collapsed. And later on, when we saw her, we did the testing for this, and as you can see, as we increase the concentration, the wheel and size of the reaction increased, and this was the 3rd case in the literature which we could found on the net. Patients like this, reacting to any drugs, leading to tongue edema, hoarseness of voice, laryngeal edema, very dangerous. They can undergo anaphylaxis and stridor anytime, and one has to be really careful for such patients. Treatment for drug allergy is avoidance, where the same drug and the family which is cross-reactive should be avoided. Desensitization is done only when alternate non-cross-reactive drugs can't be used. Here, a rapid administration of small doses of drugs are done to higher concentration, and this causes a temporary induction of drug tolerance system. Also, this makes the effector cells very less reactive to a IgE response. Why we should refer to an allergy specialist and know whether it is a real allergy or just a mislabeling. This is important because once a patient is mislabeled as a drug allergy, he carries this label lifelong. And because of this label, as we saw in that young boy, he had some seven drug allergy written by the Kasturba people, which is the only ID hospital in our city. What happens is the patient is then given an alternate drug which can be more toxic, less effective, and more expensive. This also increases the morbidity, cost, and gives a poor quality of life to the patient and adds on to the psychological stress. Like one or two patients whom I have tested was the only reason was do we really have this allergy? If we have, what is the safest drug we can go for? So this is the psychological stress which a mislabeling can give to a patient. Also, it causes a lot of antibiotic resistance because we are using higher antibiotics. It referral to an allergist will help us to identify if the patient has a true allergy. It will help us to classify the patient into low, intermediate, and high risk. And also, it is important for the patient and the family physician to be aware of such an entity, because accordingly you can educate them and tell them how to prevent such an attack. So to conclude, drug allergy is a very common clinical problem. Allergist assessment is important for appropriate diagnosis and management. The diagnosis relies on history, physical examination, skin testing, graded challenge test, and procedures. The treatment is avoidance of the offending drugs. Alternative drug with different structure should be used if possible. Cross reactivity should still be taken into consideration. And if a particular drug which the patient is allergic to is indicated and there is no alternative, try to go for desensitization. So I thank you for your patient listening. But as I say, drug allergy is an enigma and we still have a lot to learn in this subject. Thank you once again.
Thank you, Dr. Sunita. I think I can see the iceberg is much bigger below the surface than above the surface. That's what you showed us
Yes,
right
sir.
now. And maybe I request the audience to ask questions for Sunita. Dr. Purohit? Dr. Purohit, Dr. Ashav Kaur, please ask questions.
Dr. Purohit, you can type it in the chat box.
Yeah, and we'll read out.
Okay. Can desensitization itself can produce severe reaction?
Pardon?
Can you desensitize?
Repeat?
Can that cause a reaction? Desensitization itself, desensitization.
No, so the concept of desensitization is that when you give the drug in very small dosages, we can start right from 1/20th of the drug, and when we increase it, it doesn't cause a reaction. That is the concept of immune tolerance. But the only problem with desensitization is once it is done, once the patient stops first of all, the patient has to continue the drug so that he doesn't get a reaction. And if the patient stops this drug for few days, we have to desensitize the patient again for the same thing. So this is very commonly done abroad for chemotherapeutic agents if they don't have any other alternative. But then again, it has to be done in a ICU setup, or where you can monitor all the reactions and anaphylaxis also. But the chances are very, very less. That's the concept of desensitization.
Dr. Aishav Kaur, you have a question?
Good evening, ma'am. Good evening, sir. It was an amazing session. I wanted to ask, ma'am, when we do Montax or for Montax test, uh, they also put it like they do the same test on the skin. Uh, One of my friends or they said कि in doctors it is common to get a 10 millimeter diameter because we are already sensitized. Is this true?
So Mantoux test is not similar to a drug testing which is done on the skin. Okay, because see, this drug testing is very common in surgery. We do a lignocaine testing, and it's not the doctors, the sisters used to do it for us at Nair Hospital. So when you do this, you just give a subcutaneous dose of the drug. Okay, and whereas in drug testing, it is the skin prick test, which is only in the epidermal layer, and the second is an intradermal test, which is between the two layers of the skin. We don't go subcut. So when you do a Mantoux test, it is basically to see the whether the patient has been exposed to a tuberculous bacilli or not. In doctors, this is very common because whether we had an infection or not. But when we practice at a place like KEM or any other government hospital, we all are exposed to tuberculous bacillus. It is only the immunity which saves us from an infection. So
Okay,
yes,
ma'am.
you can get a positive reaction, but that is subcutaneous. It is not similar to a skin testing for
Drug.
a drug allergy. Yeah.
Dr.
Thank you so
Bakshi,
much, ma'am.
Dr. Bakshi, your question. Dr. Bagchi?
Sir, there are two more questions in the chat box, sir.
I know, but I thought Dr. Bakshi had a question. He raised the hand, but I think it's probably not the question.
Hello. Hello.
Yeah,
Am I
please.
audible?
Yes.
Actually, I want to know, I am from School of Tropical Medicine, Department of Clinical Pharmacology. Actually, we have encountered few problems like in this drug hypersensitivity and desensitization. One is that few days ago, we have come across a case of having increased intradermal hypersensitivity of an HIV patient affected by the Trypanosoma pallidum and infection. And actually, he was sensitive to the penicillin. So intradermal test was positive, and in that case, the patient was, considering it's an irritative reaction or irritant reaction, they have gone through this injection of this penicillin, and the patient didn't have any rash with the first dose. But eventually, after the 3 to 4 days after the second dose, the patient developed the generalized maculopapular rash. So they referred the case to us, and by going to this patient, we have seen that we have diagnosed it as not as a case of immediate hypersensitivity reaction, and we have advised for an alternative therapy like doxycycline. But what's your take in this regard? That if a patient is having some intradermal test positive, should we go for the skin prick test to diagnose it whether it's a definite case of drug allergy, or if the patient doesn't have any immediate type of hypersensitivity reaction, like it's a delayed type of hypersensitivity reaction, so what's the role of desensitization in this case?
So desensitization is not for a delayed reaction, first of all. The desensitization is usually for a type 1 reaction only. Now coming to your first question, I think that if you never do an intradermal test and then do a skin prick test, it's always the skin prick test because skin prick test is very safe. Here you usually take the undiluted IV form of the drug, put a drop on it on the skin and just prick it with a needle. In an intradermal test, you go inside the dermis and you inject 0.02 ml of it. But this also we never do it for the undiluted doses. We always go for dilutions. Now this, how to make the dilution, there is a protocol for each and every drug. There is something called as a non-irritant concentration for the intradermal test. So it can be 1:10 one for one drug. It can be 1:100 for one drug. Now, if the person has injected a irritating concentration, there will be a reaction because that is not the protocol for that drug. So whichever drug you want to test, you have to see the protocol, check what is the non-irritating concentration, and that concentration has to be used for an intradermal test. Otherwise, we will get a false positive reaction like you said. Maybe it was a positive thing which they have misinterpreted and so on. Now, many times if a patient has not taken a drug, the first time nothing happens because when the drug enters the body for the first time, that is the time when it gets sensitized. That is the time when the specific Ig antibodies are produced for this drug, specific drug, and these Ig antibodies will now go and sit on those receptor on the mast cells. Now, when you give the same offending drug the second time is when a Ig reaction happens, the mast cell degranulates and the patient gets a reaction. So many times the patient say, I have never ever taken this drug. How can I become sensitive or allergic to this thing? So the answer here is a cross reaction. If you have given another drug which is cross reactive, has a similar structure to that, a patient can become sensitized to that other drug also. So that is a very big hitch in drug allergy because of the cross reaction with each other.
Okay, there are just
And
there are,
many times we confuse a pseudo allergic or an anaphylactic reaction for a IG reaction. That is why the role of serum tryptase. So whenever you feel this is some reaction, always immediately take blood from serum tryptase level.
yeah, Dr. Sunil, there are two questions, and I would be happy if you can give them brief answers because I think
Okay,
there's
okay.
another session behind that. There's a question: how can we desensitize a person who is sensitive to pollen or weather?
Pollen desensitization is very well possible. These are patients who have allergic rhinitis or asthma. We do a skin prick test. Whatever pollens are positive, we give them a oral immunotherapy for that pollen, and the results are wonderful.
Okay, the next one is about from Dr. Deepak Bharambe. Multiple drug reaction. What can we and how can we advise the patient about choice of drugs if there are multiple drug reactions?
Yeah, so here you have to refer to an allergy specialist. The specialist will understand whether actually it is a true allergy or not. We have to prove that it is a true allergy, and then he will chalk out a plan for you that if you need to use the antibiotic, whether you can use that one. If not, then which are the alternate antibiotics, because here we have to check the cross reactivity of that culprit drug to other drug also. And then a chart is usually made what is the safety of other drugs for that person. So it's a huge, it's a big question, and I have tried to answer in very few words.
Yeah, Dr. Sunya, thank you very much for this wonderful presentation and patiently answering questions. I think allergy is a subject where nobody is allergic to the questions, but the time is not going to stop wait for us. So thank you very much again.
Thank you, sir. Thank you, ma'am.
Thank you, sir. With this, we come to the last session for the day. As we move ahead, I invite Dr. Dennis Xavier, sir, as the chair of the session. Sir is head, Division of Clinical Research and Training, and Professor of Pharmacology at St. John's Research Institute, Bangalore. After his MD in Pharmacology, Sir has completed MSc in Clinical Epidemiology and Biostatistics from the McMaster University, Canada. In his 25 years of teaching experience, Dr. Xavier has over 170 publications and been a part of over 40 projects, with the main area of research being non-communicable diseases. Sir has also formerly held the position of Vice Dean Postgraduate at St. John's Institute. I request you, sir, to kindly introduce the speakers and take the proceedings forward.
Right. Thank you, Dr. Palvi, and thank you, Dr. Nitya Gokte, for inviting me to this really exciting session. I've listened to all of the talks, and I think something really exciting is coming up now. So we have two sessions, but shortened sessions, 10 minutes each. Maybe that's what happens when you're the host. You sacrifice time for others. But just permit me a minute or so to just give you a quick perspective on this. Probably about 12 years ago, I was at a meeting, and then we were talking about pharmacovigilance and all the different procedures that were going on. And I'm not an expert in pharmacovigilance. My department works since the 1980s. We've been doing work, so I don't work directly in pharmacovigilance, nor do I have much of a clue in artificial intelligence. So I don't know why they chose me to be a chairperson here, but still, I'll try to do my best. So at that time, about 12 or 13 years ago, I asked people, okay, you submit all these reports, it goes, finally Uppsala, and then it comes back, and then a drug gets banned. It takes a long time unless it's something really dramatic. I asked a question: why can we not prevent, adverse reactions as much as possible in the first instance itself, and later on, as the years went by, I figured out that you need really good electronic medical records and linkages into not just the laboratory parameters, but also now currently it's also possible to link with your your gene sequence data. So all of that is coming, but before even we get there, we can use modern technology is what I've learned, and that is artificial intelligence. So for both of these, we need large data. I try to find out what is large data. Large data is defined with three V's, which is it is large volume, is high velocity and a lot of variety, 3 V's. And then in terms of quantity, what is it? So in the 1900s, if you have data of about 1 GB, it was large data. Then it progressed on to 1,000 GB, which is a petabyte, and now it is 1,000 petabytes, little over 1,000 petabytes, which is an exabyte. So these kind of data are so large. Human intervention to try and analyze that will be challenging, and it will get delayed. So humans are so intelligent, they get machines to work for them now, right? And that's where artificial intelligence works, where traditional methods of analysis is not possible. And that's where we're going to hear now from Dr. Nithya Gogte. It's my pleasure to very briefly introduce her. She's professor and head of the clinical pharmacology department at KEM, and she has also trained at the WHO coordinating center, and she and I have been part of several government of India review committees, and I really appreciate her clarity in her communication. And personally, I'm looking forward to listen to you, Dr. Nithya. And I'll just take a few more seconds to introduce your colleague, Dr. Bhaskar, who trained at All India Institute. He worked in pharma companies earlier, and now he's in academia. So he's also from KEM, and both of them are going to give us, a flavor of artificial intelligence and its important role in pharmacovigilance. And as doctors, our job is to treat patients and save lives, and this is where we come in. So over to you, Dr. Nithya Gogte.
Thank you so much, Dennis. Thank you very much. Let me just stop my video and let me see if I'm still on the learning curve of WebEx, or I am. All right. Audible, visible, all good?
All good, Dr. Nithya, please go ahead.
Yeah, so again, thank you, Dennis. And let me just start by saying what Dennis has already said. I have absolutely no expertise in artificial intelligence. I do have expertise in pharmacovigilance. So what I'm going to do over the next maybe 10 minutes or less is to take you through some of the key principles of artificial intelligence. And if some of you find this very basic, do bear with me. What I will do is to start with the historical origins of artificial intelligence, define artificial intelligence, and a term which is frequently used along with AI, that is machine learning, understand what are knowledge graphs, take you through a bit of artificial intelligence and how it helped drug development in COVID-19, and finally end with a summary of the presentation, and I'll let Bhaskar take over to talk about the applications of AI. So it was in 1956 at the Dartmouth College, where all of this began. And this was the first ever workshop to discuss artificial intelligence. And there were key people in artificial intelligence. This is John McCarthy, Marvin Minsky, Claude Shannon, among others. And what was the question that they posed? So what they said was, could every aspect of learning or any feature of intelligence, can it be so precisely described that a machine can actually be made to simulate it? So we are talking about aspects of learning, intelligence, precision, and all of this being done by a machine. So this was the first ever workshop and movement in artificial intelligence. Just give me a moment. Yeah. So what happened after this, unfortunately, was not just in this area, but in other areas, that artificial intelligence began to produce underwhelming results. And I'm going to give you one example. And this is the English sentence: the spirit is willing, but the flesh is weak. And this was decided to be translated into Russian. And this is what happened: the vodka is good, but the meat is rotten. And there was a point when a lot of funding for artificial intelligence was actually cut, and they called it the winter of artificial intelligence. So if we were to analyze what went wrong, Dennis has already talked about data. It is limited information. So they had limited information. When we say information, that's data, and limited computational facilities, right? But today, both of it is possible. We have humongous data. There are 3 V's he talked about. We have heterogeneous data, and we have super computational abilities. And therefore, the 2 key paradigms of artificial intelligence which are possible today is essentially you have the data, you model the data, and you infer from the data, right? So that's possible today with artificial intelligence. So if I were to define artificial intelligence, it's essentially any human-like intelligence exhibited by a computer, by a robot, or any other machine. So what are we doing essentially with artificial intelligence is to learn from examples and experience, recognize objects, patterns, understand, respond to language, and therefore eventually make decisions and solve problems. The associated term with artificial intelligence is machine learning, right? Now, what's machine learning or ML? It's an application of artificial intelligence which focuses on development of computer programs. But what's important is that it is intuitive. So the aim of machine learning is essentially to allow computers to learn automatically without human intervention or assistance, and therefore accordingly adjust their actions, right? So that's your machine learning for you. Just give me a moment. But the premise of all of this is essentially what I'm going to talk about, and I think that's the crux of artificial intelligence, and that is knowledge graphs. So what happens is data, whether it's in your brain or whether it's in the computer's brain, is stored in graphs that mimic intuitively the way humans understand information. And all of us humans inevitably collect, store, and what we call as join the dots, or join the information. And I'll illustrate this with an example. So this is what essentially a knowledge graph looks like, right? So let's say that there were two people who were with me when I was in Sion Hospital. One of them was James and one of them is Lily, and they went back to Paris after completing their studies, right? Now, I have been invited to Paris to give a lecture, right? So I've never been to Paris. So what do I want to do? I want to see the Eiffel Tower. So what do I remember suddenly? Oh, remember the James who was in class with me? Why don't I just reach Paris and call him up and say, I want to see the Eiffel Tower? So fine, he does that. So I've already joined the dot of Eiffel Tower, Paris, and James. Then I also remember that James is a friend of Lily. I don't know Lily that well, but I do remember her, and it's more likely that she'll remember me. And I remember that she likes Da Vinci, so why don't I ask her to take me to the Mona Lisa and see it? So what I'm doing is essentially all of the knowledge that I have accumulated, or all of the knowledge that's available, I'm putting it together, I'm connecting the dots, and I'm therefore trying to see Paris. So this is what a knowledge graph in medicine looks like. And what I'll talk about and now end with is how artificial intelligence helped us with COVID-19. And this is the Benevolent Artificial Intelligence, a company based in the UK. Now, we're familiar with COVID-19 now. So you have this, second, let me just use the pointer. Yeah. So you have this SARS-CoV-2, the virus, which binds to the ACE2 receptor, which is expressed in the AT2 lung cell, right? Then there is a process of endocytosis and there is viral entry. Now, so you want to block this viral entry and you want to address this endocytosis. So how do we do that? So you already have the knowledge graph with you. You already have information with you. So what you do, you look at the mediators. So you have this kinase AAK1, which mediates or promotes endocytosis. You also have the cyclin G-linked GAK, which mediates endocytosis, right? So let's look at fedracitinib, sunitinib, erlotinib. Perfect. So they block AAK1 and therefore they could be potentially used for COVID-19. But then you have another drug, which is baricitinib, which not just impacts AAK1, but it also affects GAK and therefore endocytosis. But more importantly, because it's a JAK1/2 inhibitor, it's actually going to impact cytokine signaling and therefore it's going to address your inflammation. So herein you have a potential drug which is not just impacting viral entry via endocytosis, but is also impacting inflammation. So it could be used not just for mild moderate disease, but it's also going to impact severe disease. So this is baricitinib. And this was an early paper that was published in the Lancet when baricitinib had not been used for COVID-19. What did they say here? By reducing both viral entry and inflammation in patients, baricitinib could potentially be used as a therapeutic option for COVID-19, right? And if you look at this graph here, This paper was published sometime in February 2020, and the US FDA actually accorded approval for baricitinib, which went on to be used along with remdesivir in the Act 2 study. So the approval is November 2020. The early signs from artificial intelligence came in February 2020. So to summarize, what artificial intelligence, machine learning really is how a computer synthesizes knowledge, synthesizes information, models it, and it helps us to make inferences. And a lot of it is going to be transformative. The question is, as of today, is it transformative for for pharmacovigilance? We learned this from Dr. Bhaskar. So I will stop my presentation here and hand over to Bhaskar, and maybe both of us can take questions at the end of it. So thank you, Dennis.
Thanks, Dr. Nitya. So we'll have Dr. Bhaskar's slides up.
Hello?
Yes.
Yeah, just give me a couple of minutes. I'll share my slides. Yeah, it is visible.
Yes, visible. Please go ahead, Dr. Bhaskar.
Very good afternoon to one and all. I thank the organizers for this opportunity to allow me to speak on artificial intelligence in pharmacovigilance. Though just like Nitya madam, this is also not my area of core area of expertise, but nevertheless, I'll still try to give a brief overview as to what is artificial intelligence and how it is being used in pharmacovigilance and how it can be used. And I'll try to keep it simple. I'm not going into significant detail. I'll explain what, why, and how of artificial intelligence in pharmacovigilance with some examples which are available in the literature. I'll mention some challenges and concerns, and I'll summarize my presentation. So what exactly is this artificial intelligence? In the previous presentation, Nitya madam has already given the definition as to what it is. But if I want to understand in very simple terms, I can say that, let me take an example of a, say, surgery resident. A surgery resident, when he or she gets into the residency, in the 3 years, the resident will undergo training. And at the end of the residency, he or she is expected to develop this intelligence. This intelligence basically refers to the ability to take a decision as to whether to operate on a patient or when not to operate or what treatment is to be given. So the ability to take a decision is what a resident learns during training. In the same way, if I train a computer software so that the computer software is now able to take decisions, that is what is artificial intelligence. So why artificial? Because here the student is a non-living thing. It is a software. And how do I train it? With the help of data sets available from the real world studies. So whatever existing literature, whatever data sets that I have, I can use it to train the software, and so that now the software is capable of taking decisions. That in a nutshell is artificial intelligence. So why do we have, or why is this artificial intelligence being considered in pharmacovigilance? Because of the speed at which it works. The detection of adverse events can be faster if artificial intelligence is used. And with this, the dosing recommendations that I'm able to give can also be faster and more accurate. Naturally, we know that a software works faster than a human. And nowadays, with the advent of this quantum processing and DNA processing, the processing speeds of the computers have also gone up significantly. So the work that can be done can be extremely fast. And as previously mentioned also, the data set that we have today is very voluminous. Now, this represents the individual case safety reports received by the US FDA adverse event reporting system. If you compare with what was there, say, 20 years ago, and the volume of data that is received today, there is a significant increase. And it is extremely difficult for humans to deal with such large and such voluminous data, right? So that is why we take the help of these machines so that the analysis of these large data sets becomes possible and easier. And with this, the decision making is improved. And of course, an attempt is made to reduce the human error. Again, as we know, softwares are less likely to commit errors as compared with humans. So we do consider that the errors can be reduced. So these are the various advantages that the artificial intelligence has in pharmacovigilance activities. So what all can it be used for, or what all is it being used for? So basically, the primary use so far is for the processing of the individual case safety reports. The artificial intelligence can be used to extract the data from the source documents and the initial classification of this data, then to see whether there is any duplication of reports. If the same case or the same report has been submitted twice, then the duplicate reports can be deleted. Then whether the minimum regulatory reporting requirements are met. So the software can check for incomplete reports, whether the particular 4-5 fields which are mandatory, they are filled or they are incomplete. Then the triage and initial assessment for seriousness. So whether the adverse reaction is serious or not serious, that classification can be done. Again, whether the adverse reaction is expected from that drug based on previous data, or it is a new adverse reaction that is being reported. That is a potential signal generation. This can be considered. Of course, data entry can be done. Causality assessment. There are softwares developed, which I will discuss in the subsequent slides. Coding of the adverse events into standard terminology, into terminologies, the classification and coding can be done. And of course, writing of the narratives of the individual cases. So these are all activities wherein artificial intelligence is actually being used in the present day scenario. So to cite a few examples from these literature, this is a software that has been developed by Roche, modified Naranjo causality scale for ICSRs, also known as Monarch-C. Now, this scale consists of 9 particular parameters which have been incorporated into the scale or into the software. And these 9 parameters are the ones which we normally use for causality assessment. Like, is the adverse event consistent with the drug use? Are there previous reports? Is there temporality? Is it explainable by the mechanism of action, de-challenge, re-challenge, and so on? So considering these 9 parameters, this software has been developed, and it has been compared with human assessment of the or human causality assessment. So this table shows the results, and the agreement. The agreement as shown by the kappa score is 77% or 0.77, which is a good figure. Also, the area under the curve is also good for the software, and the specificity in particular is on the higher side, and the sensitivity, positive and negative predictive values of the software are also good for performing causality assessment. And they have also calculated an F1 score of 0.71. So what is this F1 score? Basically, it is a balance of recall and precision. Recall, which is given by the sensitivity, and precision, which is given by the positive predictive value. So this is the formula to calculate the F1 score, which the authors have used. And using this formula, the F1 score is derived. The significance of this F1 score is, higher the score, we consider that the system is more automated. So naturally, a fully automated system will have a F1 score of 1, which is of course a very ideal software, but it is very difficult to attain. So the closer that we reach to 1, we can say that we have a automated system that is working. Another study that has compared a software with a human causality assessment. The columns here represent the assessment done by the expert, that is the human values, human scores that are given, whereas the rows here are the Bayesian network causality assessment. So if you see, the diagonal is the one that is showing the agreement, and this agreement is actually more for the higher causality ratings. That is, if the rating is definite or probable, then the agreement between the artificial software or the artificial intelligence software and the human causality assessment is higher. They have also calculated the precision and recall for these individual categories. And they again say that the precision is better for the higher causality assessment, higher degrees of causality assessment. Also, as expected from the machine, the time to assessment is also reduced. So it was a median of 4 days was needed for the Bayesian network to carry out causality assessment as compared with 8 days for the experts. Now, this is another study where they have evaluated an artificial intelligence system for assessing the seriousness of the reaction. So whether to classify whether the reaction or the reported event is serious or not serious. So that they have considered various data from post-marketing reports, from solicited reports, and from the available medical literature. And they said that the accuracy for the classification of seriousness is quite good, almost more than 80% for all kinds of data. And even the precision and recall for each of these is also on the higher side. Similarly, they have also again calculated the F1 score, and they have reached once again values to the north of 0.7. So they say that, okay, yes, this particular software can be used to assess the seriousness of the reported events. So these are some of the efforts, or rather some of the measures that have been taken by the US FDA. Starting from 2011, they have developed this natural language processing program for extraction of clinical features and for a specific case definition. They have also used these programs for identifying reports of similar medical condition, for extracting temporal information, and so on. So there are various areas wherein the US FDA is also making efforts to incorporate these softwares for the in the pharmacovigilance activities. Now, coming to what are the challenges which are faced by using this artificial intelligence. Of course, the data input has to be in a structured format that is tailored to that particular software. It has to be entered in a particular format. We cannot enter as it is, and therefore the data entry should be done very meticulously. Now, just like most of the softwares, this also works on the principle of GIGO, that is garbage in, garbage out. Meaning, if the data that is input into the software is of bad quality, then naturally the output that is going to come out is of bad quality. So the quality of the input data needs to be ensured when we are feeding in the data into the software. Again, different softwares are developed in various parts of the world. So a harmonization is also an important consideration, wherein the softwares in different parts of the world should use or should deal with same terminologies or same principles. There are privacy concerns, of course, just like any software. There is a concern that the data may be used for other purposes other than pharmacovigilance. And of course, there is a initial reluctance and resistance to the use of not only artificial, but any kind of software. So whenever there is any new thing that comes into picture, we are reluctant initially to go about it. And of course, we do watch a lot of movies, and we are very afraid. Okay, sir, how can we trust artificial intelligence? We have seen these various science fiction movies wherein these softwares or the algorithms, they tend to they tend to wreak havoc, they kill people, they take over the world. So what if something like that happens? So how can we trust this AI? So to alleviate some of the fears, I quote what Dr. Vivek Ahuja had said. This slide is taken from Dr. Vivek Ahuja's presentation in one of the previous advanced level training workshops. So what he had pointed out that we are already trusting artificial intelligence with our lives. And the example that he gave was the aeroplane, where this is the view that is from the cockpit of an aeroplane. And if you see, the aeroplane or the flight uses artificial intelligence while flying. There is something called as autopilot, which is used. When the pilot is not in the seat, there is an autopilot which tends to fly the plane. So over there, if anything goes wrong, then it can actually be life-threatening. So if we can trust artificial intelligence with our lives, then definitely we can trust with the pharmacovigilance data at least. So once again, Dr. Vivek Ahuja had said in the same presentation that we are presently at an inflection point. So we are at a scenario where the computing power is going to go up and artificial intelligence or softwares are going to be developed. So yes, it is up to us. We can make the best use of it. And one of the ways forward that has been mentioned in the literature is something called as augmented intelligence, which is considered as not a complete alternative, but something that can go with artificial intelligence.
Dr.
So,
Bhaskar, if you could conclude in 1 minute more.
yeah. Yes. So this augmented intelligence is nothing but a hybrid system where a human and a software are going to work together. So rather than having a completely automated system where the software is going to take all the decisions, the human being is going to be in the loop and ensure that there are quality checks and the performance level of the AI is what is needed. So this is what is also known as human in the loop, where this is not a completely automated system, rather a human being is a part of the system and he or she is taking care that the AI is working in the way that we want to. So this hybrid system or augmented intelligence is what is considered as one of the methods to go forward with using this technology. So to summarize my presentation, artificial intelligence is a computer software or program or algorithm that can be trained to take decisions. It enables handling of large databases and that too at a faster rate. It has been used for causality assessment and assessment of seriousness of adverse events in pharmacovigilance. And of course, there are many other applications also. The F1 score for a software indicates a balance of precision, that is the positive predictive value, and the recall, that is sensitivity. It has its own set of challenges and concerns, but it is already a part of our lives as we have seen. And augmented intelligence, which a hybrid human and machine model, is considered as one of the ways forward. So I thank you all for a patient listening, and any questions I'll be happy to answer.
Right. Excellent. Thank you, Dr. Bhaskar, and thank you, Dr. Nitya. Both were nice, crisp, and clear presentations. It gives us a little orientation into the what artificial intelligence is and its applications. So I'm looking out at the chat box for any questions. So please put a short question there in the chat box. And if you prefer to ask a question orally, please put up your hand and we'll unmute you so you can speak by then. As your questions are coming up, I have just a brief comment on this last two talks. So, well, this is what is expected of KEM, a premier clinical pharmacology institution in the country, to set high standards, to be ahead of the curve in doing this really interesting advanced pharmacovigilance seminar that we had, or webinar that we just had. So thank you, Dr. Nitya, firstly for in a very simple way telling us what artificial intelligence is. The analogy or the example you used about wanting to go to Paris and seeing Mona Lisa, that is ML, and through that you gave us another ML, which is machine learning. So that was really nicely put, and then gives us, I mean, at our level, we need to understand that concept. So that was really wonderful. And then Dr. Bhaskar, you took us nicely through its applications and also with examples, and particularly the F1 score is, I'm sure, like for me, it was new to many people. So we learned more from there. That table that you gave us from the FDA since 2011, about close to a dozen different applications. I'll kick off the first question while the others are coming. Through this afternoon, I was hoping to look or learn more about how we can prevent ADRs, preferably primary prevention. Well, maybe primordial prevention a little bit later, but that's where huge data will help us, particularly with the genome sequencing becoming so inexpensive now, and with precision medicine coming in, that's where we're going to go next. But before that, I'd like to, maybe 5%, 10% of ADRs could be prevented using the current technology. So any thoughts from you, Dr. Nitya first, and then Dr. Bhaskar, while I look up the chat box for more questions.
So thank you, Dennis. It is important, I think, to prevent ADRs. And several years back, I think almost 2 decades back, we'd actually done a study where we looked at admissions to the emergency services, and we found that 60% of the ADRs were what we call potentially preventable. And this was when malaria was very high, and something as simple as, taking an anti-ulcer medication before you give chloroquine, that was not being done. It was not that it was not prescribed, but it was not communicated. I think Dr. Sachin also alluded to this prevention with his patients. Since the topic is on artificial intelligence, I have looked up 2 narrative reviews and 1 scoping review on whether artificial intelligence can actually predict adverse reactions, and therefore you actually counsel patients. But if you look at that review, there are only maybe 3% of papers among the 100% we've actually looked at artificial intelligence preventing adverse reactions. So the bulk of the work on AI is essentially identifying safety signals, but less towards prevention. So I think it's important that at least in the context of AI, we have not yet moved towards prevention. Yeah. Over.
Right. Thank you, Dr. Nitya. But I'm sure that's what's brewing and should come out soon, very soon. Yeah. And yeah, talking about brewing or rather distilling, your analogy with that vodka was very interesting. These are things that you remember, and that's where artificial intelligence, can become a little uncomfortable. Right, Dr. Bhaskar, your thoughts on prevention and where we are with. Simple clinical, acumen can do that, a proper history, looking at the other drugs that they are taking, looking at your, biochemical parameters, your renal function tests and your liver function tests. We will do that. But, sometimes you're so overwhelmed, particularly in a hospital like KEM or other government hospitals, it's difficult to do. Can machines help us? So, Dr. Bhaskar, your thoughts?
Yeah, I agree with what Nitya Madam has said, that the available literature as such is actually very, very deficient on the prevention aspect, because most of them have actually focused on the assessment and the causality and those parts. But yes, considering that there is a tremendous scope, because once we are able to predict the incidence or predict the occurrence of an adverse event or an adverse drug reaction, definitely we can explore the prevention. So yes, as of now, all I can say is yes, there is a lot of potential, and there is a lot to explore in this area.
Right, thank you for that. And Dr. Bhaskar, could you explore if you can have your video on? It'll be nice to see you when you speak. So let's look at the chat box. So there are a few questions. There is one question on, can you elaborate on the F1 score? For someone like me who has never heard of it before, I understood it a little bit. So what we'll preferably not do is, because Dr. Bhaskar, you did a good job, so we can request Dr. Chiranjeev to look it up a little bit more, or we'll send you the references that you showed there. There's also a request whether these slides can be shared. I'll leave it to the organizers and the speakers to respond to that, maybe on the chat box or later. Of course, there is a lot of appreciation of this being an awesome, informative session. I completely agree with that. Right, there's a question on robotic surgery and augmented intelligence beyond the scope of this discussion. So I'll take my privilege of being the chair to defer that question. And Dr. Nitya reminds us there's a nice course on artificial intelligence on YouTube done from Stanford University. We can look that up. Right. Any other specific questions? Okay, I don't see a specific.
There is a separate question which asks that should medical professionals hold a qualification in artificial intelligence and machine learning?
I saw that again. Folks, we got to focus on pharmacovigilance here. Let's learn more about, because there is so much to discuss. But maybe I'll ask Dr. Nitya to respond to that.
No.
Good. That was the sweetest.
I think we need to understand the principles. Yeah, we need to understand principles. We need to understand that it's an expanding world over there. There is one person that I know who did his MBBS and MD in pharmacology, and who does only artificial intelligence now. So he mines data, he mines pharmacovigilance. So I think if you're really interested, it becomes a subspecialty today. We should know enough to understand, but I'm not sure we should have, intense training. I don't think it's as straightforward as these talks have made it out to be. There is a
Yeah,
fair
absolutely.
complexity. Yeah.
Yeah, absolutely, Dr. Nitya. I completely agree with that. So, as doctors, we need to be aware and kind of accept these things rather than saying what the hell is going to happen. And this is here to stay, here to grow, and here to make our lives better. So we need to understand it. And the beautiful analogy that you gave is really nice. And you took a real life example and then told us how it works with, and how COVID-19 drugs were discovered and how we got it from, and it would take us years to figure out if we did it the traditional way, isn't it? I mean, drug for rheumatoid arthritis, mechanism of action. Oh, there is some connection here, and machines are able to do that. And we're talking about huge amounts of data putting that together. Right. Any other questions or comments? I don't see any further questions. I don't see hands going up. So I will conclude here by thanking both of the speakers, abbreviated versions of or abbreviated, presentations. So thank you very much. It was nice and crisp and clear. And I think the purpose of seminars or webinars such as this is to maybe clarify a few things, but more importantly, to make us interested, motivate us to go and learn more and to accept these technologies which are here to say. So thank you very much, Dr. Nitya and others who organized this excellent overall workshop. I listened to everything. I blocked off my whole afternoon and I listened to it, and it was a great learning experience. So I think with this, I'll hand you back to the organizers to conclude these sessions.
Thank you, sir. With this, we come to the end of our sessions. We hope the talks have been fruitful and will take your understanding of pharmacovigilance ahead. I now request Dr. Bhaskar Krishnamurthy, sir, Assistant Professor, Department of Clinical Pharmacology, KEM, to present the vote of thanks and close the workshop.
Yeah. Thank you, Palvi. I have here the opportunity of providing the vote of thanks. Thanks as a word conveys a multitude of emotions and feelings of gratitude, respect, humility, and a sense of satisfaction and fulfillment. At the outset, I thank the Indian Pharmacopoeia Commission, Ghaziabad, that is Dr. Jayaprakash and his whole team, as well as members of the core training panel for providing us the opportunity to conduct this advanced level training workshop under the aegis of Pharmacovigilance Program of India. And it is because of the support extended by your team that such programs are made possible. I would like to thank our Dean, KEM, Dr. Sangeeta Rawat, for her constant support and encouragement to all the activities of her department. She had actually expressed her desire to attend today, but unfortunately, due to other commitments, she could not. I also thank the other heads of various other departments who, in our institute, who contribute to the pharmacovigilance activities of our department. I would like to thank the chairpersons of today, Dr. Nilima Kshirsagar, Dr. Arun Bhatt, and Dr. Dennis Xavier, for taking out their valuable time to chair today's sessions and share their valuable experience with all of us. I thank the speakers of today, Dr. Sanish Davis, Dr. Hasmukh Jain, Dr. Sachin Dharwadkar, and Dr. Sunita Shukla for their excellent talks that have surely benefited all of us. I also thank the Regional Resource Center, KEM Mumbai, that is the team of Dr. Geeta Natraj, Tushar, Badal, and others for their technical support for the program and ensuring that everything goes on smoothly. And last but not the least, no show would be a success without the audience. So I thank my lovely audience for attending today's program and actively participating in it with your questions and making it a huge success. So thank you and see you all in the next ALT program.
I'd like to wish everybody a Merry Christmas and Happy New Year. Yeah, bye-bye.